A very good morning to all of you, and I'm sure you had a great. Hi, sir. Can I request all the dignitaries to please have a seat in the audience and we can begin the program? Yes, sir. So, very good morning and welcome back to the second day, first session, which is Alumni Talk Series. I'm sure you must have a great time yesterday during the I think there are a couple of event panel discussions and cultural event as well as the alumni seminar series, and you could reconnect with your old friends, colleague, and the people over here who are continuing their journey here. So now I warmly welcome you all to the second session, which is the Alumni Talk Series 2. And in this session, we will feature a three short talk by esteemed alumni, showcasing their journey, as well as they will be presenting a diverse perspective from the academia, industry, as well as the translational research. So we will have a three talk. First, we will begin with the Professor Pankaj Seth, and then we will have Dr. Jha, and then Dr. Thakur. So, to chair this particular session, I now request our session chair, Dr. Dinesh Kumar Dikshit and Dr. Rashmi Singh, to be seated on the dais to begin this particular session. So, I welcome you, sir and ma'am. So I'll give a brief introduction of our esteemed chairperson today for this particular session. Dr. Dinesh Kumar Dikshit, he doesn't need any formal introduction as we all know him, and he had a very long, glorious career at CSIR-CDRI, and he served as a faculty member in medicinal chemistry and process chemistry division, and he got superannuated from the medicinal process chemistry division itself, and he has published numerous publications, and his work has been cited widely, and that reflects his long lasting career in the pharmaceutical and chemical research in India. Our co chair, Dr. Rashmi Ma'am, she is currently serving as a professor in the Department of Geology at Banaras Hindu University, and she completed her degree in 2000 from here, and she has done couple of postdoc. First postdoc from NNI New Delhi, followed by the Case Western Reserve University and Oklahoma Medical Research Foundation. So we are delighted to have you here, and we are thankful that you have requested our request, and now you are chairing this particular section. Now I formally request you to initiate the section, and I now request all the presenter to adhere with the time of 10 minutes so that we can smoothly run this session. Thank you very much.
Thank you. Welcome you all. I think it's a very cold day, just like yesterday, but the lectures will keep you warm. And I will invite now the very first speaker of this session, Dr. Pankaj Seth. Dr. Pankaj Seth has done his PhD from pharmacology under the guidance of Dr. Shrimal and Dr. Madhu Dikshit. And since then, he has moved on to NBRC, where he is presently a senior professor at Manesar, and is a leading neuroscientist specializing in molecular and cellular neuroscience, neurovirology, and neurodegenerative disorders. His research on virus-induced neurological damages, including HIV, Zika, and SARS-CoV-2, using advanced human neural models alongside active mentoring and national research and leadership. He is a fellow of several academies like INSA, NASI, and fellow of Medical Academy of Sciences, India. Now, Pankaj Seth.
Thank you so much for your kind introduction. I think my biggest introduction is that I'm a product of CDRI, and I did my PhD under Dr. Madhu Dikshit's guidance. So my teachers are listed over here. The first one is, of course, Professor B.N. Dhawan. I grew up in a family of scientists, always since my childhood when I was in school days. People who would come to my house for dinners and lunch and breakfast were all international delegates who were coming to Indian meetings. And of course, we had Dr. Nityanand, Dr. Dhawan, Dr. Shrimal, and Proserpine Tandon and so on. So I grew up as a scientist right from the beginning. Of course, as Madam said yesterday, I wanted to be a medical doctor, which my son is now fulfilling that dream. But I could not make it to KGMC or any medical college for that matter. And then I took up this profession, which I'm very proud of. And thanks to the guidance and the foundation which was laid at CDRI. So my journey as a scientist started as a summer trainee at Department of Pharmacology, and I went to there while I was doing my final of BSc from LU. And Dr. Dhawan put me with Dr. Rakesh Shukla ji to learn the blood-brain barrier model, and I worked with Dr. H.K. Singh on Picrolive. And I'm very proud to see yesterday that the Picrolive is in the phase 3 at that time. So then, of course, the journey started. I completed my MSc from Lucknow University Biochemistry. I was a topper there. And then was selection of guides. I was lucky, and as Dr. Prasad was saying yesterday, he had 2 guides which were very difficult, and he was trying to make a balance between them. I never had that problem because I was so lucky to have these 2 wonderful human beings and great scientists who would make the best out of me. So I worked between ma'am and Dr. Srimal, and I finally completed my PhD and then went for my postdoc. But today I'm going to talk to you about what we have been doing recently. Since it's a scientific talk, my my slides will have some scientific content. So the lab is trying to understand how viruses do damage to the brain, how do they get into the brain, and then what do they do to the brain cells. So the lab has been particularly interested in understanding how HIV, Zika virus, and then now SARS-CoV-2 and dengue, what are they doing to the brain cells. And the model system which we have started to work with was the human fetal brain derived neural stem cells. And the reason for that was because we wanted a cell type from human origin, because we started working with HIV, which was a human specific virus. And that's how we started to work with this model system, thanks to the training at NIH which I had during postdoc. And we routinely make these human fetal brain derived neural stem cells, which are primary in nature. They are not cell lines, but they are primary cultures, and this is a great advantage over them. We do a lot of things to them and finally get something which we believe are neural stem cells. And we run a battery of neural stem cell markers such as Nestin, SOX2, Ki60, and many, many more. And the beauty of this model system is that it grows not only as monolayers, as is seen over here, but also as neurospheres. So we work with 2D and 3D cultures of human fetal brain derived neural stem cells to understand how brain cells or the viruses or the viral proteins affect different kind of brain cell types. And we published several papers from the lab as well as from others, which have been featured in different various forums, including Nature India and International Society of Neurobiology. Now, what is the potential of this system? It's immense. We have actually understood how HIV affects the stemness of neural stem cells. Because there are stem cells, we can work at 3 angles. We can work at the neural stem cells itself, or when they're differentiating into brain cell type, because they have the potential to differentiate into astrocytes, neurons, and oligodendrocytes. And having said that, we have studied the effect of HIV on neurogenesis. We have studied how HIV is affecting astrocytes, which is one of the most important cell types. And this journey continued, and the papers started pouring in, resulting in a significant contribution to the field to understand how viral proteins and viruses are affecting brain cells and at what steps. So we dive deeper into the mechanistic aspect, and we always went for the difficult task to understand the mechanisms, how viral proteins were affecting brain cells. So we then entered into Zika virus, and I'm going to talk to you about Zika virus mainly because the topic is about neurodevelopmental disorders. So what does Zika virus do? If you can see the slide taken from CDC, you'll see that there's abnormally small head size of infant which are born to mothers which were infected with Zika virus. Now, there was strong correlation over here because this was happening in only the first trimesters. So if I could, so if you see over here, if a Zika infected mosquito bites a woman in the first 3 months of her pregnancy, it is likely that the child is going to have something called as microcephaly. Micro is small, cephaly is head, an abnormally small head size of an infant which is born to mothers which are infected with Zika virus, 5 to 10% of such infants will have microcephaly. And mind it, it is only if this mosquito bite is happening in the first 3 months of the pregnancy, in the first trimester. So there's something happening in the first 3 months which is not happening later. And that is a robust proliferation ability of these neural stem cells, and all the head circumference and everything is dependent on the first 3 months of the development of the infant. And that got our attention. Because we had these human fetal brain derived neural stem cells, all we had to do was put the virus or the viral proteins together to see if it is really happening and they're targeting the neural stem cells. Literature by that time had suggested that yes, it does, but we wanted to again understand the mechanisms how that is happening. So what we did was next slide. Okay, thanks. So what we did was we looked at the structural and non-structural protein of the virus, and we then transfected them, overexpressed them in our neural stem cells, and then tried to understand if it was affecting it was doing something to the neural stem cells. So what Reshma did and was that when we have these fetal derived neural stem cells and we overexpress the E protein, which is the envelope protein of the virus, we found that it was affecting the proliferation because it inhibited the growth of the neurospheres as compared to the empty vector. We also understood the deep mechanisms and we found the microRNAs which were targeting the pathways for neural stem cell differentiation and neurogenesis. I would need an extra 1 minute only. This works. Yes. So then we tried to understand, we published several papers again to understand how Zika virus was affecting the proliferation and leading to the premature neurogenesis, which was also faulty. So all this was actually affecting the development of the first trimester. Okay, so this is what we found. And then once we published all these papers, again there was another student who was very enthusiastic and asked the question in a regular lab meeting that why don't we, sir, look at autophagy also, because there's a possibility the Zika virus is affecting the autophagy. And why autophagy was because it affects the Wnt and the notch pathway. Okay, so we looked into that, and what we found was that autophagy was affecting the proliferation as well as the differentiation, premature differentiation, because it was affecting the pathways for the Hess pathway and the neurogen pathway. Okay, so all this had a bearing on neural progenitor cells. We published this paper and moved on to understand. Am I doing something wrong? Okay, forget it. So then again we moved to the more complicated systems because we wanted to understand this cellular architecture as it develops in the utero. Now, many people are developing organoids. So it's not that we also started doing it. There was a need in the lab to develop this organoid system. And since we are at National Brain Research Center, our focus was to develop human brain organoids. And one of the reasons, I'm sure you could have seen this slide very recently, which came just in September 25, is that even NIH in these difficult times of funding has decided to put $87 million in 3 years to establish nation's first, which is US first, dedicated model system for brain organoid. So we started doing that also much before this, and thanks to the Scientific Advisory Committee meeting which happened in 2024, in 2025 January we had some extra funds allocated from the institute to develop this organ model system. Where does it start from? It starts from iPSC. You need, again, embryonic stem cells. We took iPSC cells, run them through all the pluripotency markers, and once we confirm that they have all the pluripotent markers, 2 of these are Yamanaka factors, which you can see over here, and then we wanted to make it more complicated. Why? Because we want to understand how our viruses or pathogens are doing to the brain architecture and developing early events of the brain. So we included microglial cells into them. Now this is where our brain organoid system is different than others. So usually you will see cerebral brain organoids which are devoid of these, because the iPSC cells, when you put them into a neuroectoderm origin, you'll usually have something which will have all brain origin cells. Now microglia cell is not a brain origin cell, it is a resident cell. So you have to include it. So what we did was, we took iPSC cells, we push them into my hematopoietic lineage. So this is happening separately at day -12, and we start that, and then we start our organoids, and then integrate and get this system to start with. We start with embryonic bodies, which you can see over here. We use agrivels, which helps us to give very uniform organoid system. One of the challenges which is there is, you have different batch variations and size variations. We could overcome that with using agrivels. This also gives you an advantage to have up to 1400 organoids in one well of a 12-well plate. So this is the robustness of it. Scaling up, we also did the Indian Jugaad system, which was cheaper. We used the low adherent 96 U bottom plate and had the organoid system, which were very similar to the somewhat similar to the agrivels. So it's a recipe-based, kit-based assay, which is actually seems easy, but it's not. But if you do it right, you can grow them up to many months. We have grown them up to day 90, 3 months, which is enough for our time points. And you're seeing some pictures over here up to day 50. Now, very quickly, this is how it looks. This is the size. If you can see those white rye-like or mustard-like appearance, it's up to 1 to 2 millimeter. They can also make assembloids. You can make different parts of the brain and bring them together. This is what the field is doing. But our microglia are again different because they had, our organoids are different because they also have the microglia cells. Now, what are the reasons why it is important? Please, I want your attention only to this part of it. The pink cell, pink portion of this is replicating that. It is replicating what is exactly happening in utero. So this is a 9-month pregnancy of human fetal brain development, and up to second trimester, everything which is happening in utero is reflected in the organoid models. And this is why it is important for studying the early brain development events. It also have all these markers which overlap in different cortical layers which develop in the brain, and we looked into that and look at, see beautiful rosettes which are PAX6 positive, which are basically important for NPC or the neural precursor cell identity. You look at DCX, which is early neurogenesis. You look at CUX1, SOX2, and all these layers, when they're supposed to be appearing, were appearing in day 30 and day 60. And this last 2 slides, this also further confirmed that these day 60 organoids also had the cell types and the layers which were there supposed to, again replicating what was happening exactly in utero. And once we were confirmed with that, we asked these questions. Again, I would like to draw your attention to the SARS virus now. There are some reports which are suggesting that they have a bearing on the neurodevelopment. So what we did was okay, so we have these organoids, day 60, we put them in with virus at MOI 1 and 2. We do these experiments with RCB because we do not have a BSL-3 facility. And so then what we found was the important markers such as relin, which is for neuronal migration, DCX, again, early neurogenesis markers, and other layers were also affected, which is basically next, please. Next. So just to summarize, that these fetal brain derived neurostem cells are a wonderful model to understand early development. We have gone from 2D to 3D. We have these organoid culture systems which are giving the complexity of glial and neural cells developing in a time-wise manner. And then when you have virus into them, next kar dijiye. And we have many of the work which we are doing with COVID, we have also tested with the human postmortem tissues from Nimhan's brain bank. So we validated what we find in petri dish is actually happening in patients. Next one, please. These are the wonderful people who have made it possible in my lab, except there's 2 in the middle, And thank you, CDRI. You have laid the foundation for me. It is very important. And next one, the CDRI has given me so much in my life, including my next one, please, life partner. And thank you for inviting me, Dr. Yadav, particularly for squeezing me in. And I'm really very thankful to the audience to be here. Thank you so much. Sorry for overshooting the time.
Now I will invite next speaker, Dr. Jha. I take liberty to introduce him because he is a product of medicine chemistry department. He did work with Dr. Kanchan Hajela, if I am right. Since then, he moved on doing several postdocs and ultimately joined the industry where he is doing very cutting edge research. And his formal introduction is given here. He is serving as a head of technical operations at Cohance Life Sciences, Hyderabad at the present. His company is leading complex chemistry manufacturing initiatives. Expertise spans API, specialty chemicals, CDMO services, drug discovery, and organic synthesis. You realize working in industry is very different than what we do in academia. So he will possibly throw some light on that. Mr. Jha.
Thank you, sir, and thank you for a kind introduction. Good morning, everyone. I just wanted to share one quick story. When the first came that CDRI Alumni Association, I got two quick messages. One from New Jersey, Dr. Mahavir Prasad, and one from my CEO, Dr. Sudhir Singh, with the link that you need to immediately register. Unfortunately, both of them could not come here today, but that's how our excitement when we heard about the CDRI Alumni Association. Especially in my career, the role of both of the CDRI alumni has been immense. After completion of my PhD from here, I have been mentored by alumni. Dr. Sudhir Singh, from the very beginning, he mentored me, and I have introduced to Dr. Mahavir Prasad when he was my client at Novartis, and when the first thing he heard, Ashok, you are from CDRI. And that's how our relationship started, from alumni mentor to a very good friend, and so happy to be here. Today, my topic of, my title is little different than the regular scientific talk. I will be talking about a little different world, creating a lot of impact, but highly underrated. So, would like to share some of things that what we can do. So, Indian pharma CDMO, how we are enabling and how we are helping accelerating the drug research and development. Just wanted to showcase some of them. But before I go, it's only fair because the name Cohance sounds very new. Okay, where is Cohance Life Sciences? This has started with the strategic acquisition from 2020. 6 companies have been merged, strategic acquisition, starting from RAKM Pharma, ZCL, Avra Laboratory, very well known and founded by Dr. A.V. Ramarao. Suven Pharma is very well known in India for over 3 decades of legacy in commercial manufacturing. And Sapala Organics is one of the unique CRO for last 2 decades in the nucleic acid chemistry. And recently in NJ Bio. NJ Bio is based in Princeton, New Jersey. Have been some of the best CRO last 4 years in the area of ADC. So our philosophy have been very clear. We wanted to do a technology platform driven, build in CDMO. 3 major in terms of pharma CDMO is a small molecule, ADC, and oligonucleotide. And our vision has been very clear, from a discovery to commercials. So for a small molecule, we are not doing in a discovery segment, from development to commercials. From an ADC, we have strategically acquired that all the CRO, like from an early bioconjugation discovery to the preclinical up to the phase 2, comes from NJ Bio, and from India side, we're in Avra, where we have a commercial payload manufacturing. So that's how we have integrated. 3 major segment. Pharma CDMO is a major segment we have. Then the API plus and specialty chemicals, and we have worked all across the globe. So I thought it's only fair just to say the Cohance name is come only. It's a fully integrated this year itself in May when the Suba and Cohens all joined and become one entity. So all the 6 companies become now in one entity. It's called Cohens Life Sciences. Very clear vision: collaborate, enhance, and deliver. Okay, so this is the one thing. Whenever we say CDMO, okay, it's oh, what is CDMO? Is this like a contract service, or maybe vendors? So because this has gone through a long journey. This has gone through a long journey from an early 1990s. It was more sort of a cost-effective alternate, or say hired for capacity. That's how it started its journey. Till 2020, it become an alternative for an in-house capability. So okay, you can work with the CDMO. You can have they have expertise. You can hire for expertise. But so many there have been paradigm shift 2020 onwards. It's like in partners, collaborators, where it's like because they can support from an end to end. When we say concept to commercials, from a very early stage of a target validation to helping in the launch of the molecule in the market. So it has gone through complete change from a vendor, contractor to service provider to a partner. So that's how it's impacting. Like to share some data. I think this is always only fair. You can see that it's up to 185 billion dollar in 2025, and projected to go double in another 10 years. So that's a very huge market. And why? You can also see here from 2014 to 2023. Even this data has been recently published from all the large pharma. So it's in-house versus outsourcing. So roughly 50% of the whole drug discovery and development portions are outsourcing. So this is a huge market, and it happens because it's ability of the CDMOs to work in a one-stop place. So the one-stop solution from an end-to-end services. So they have really picked up their game, how they can go from an end-to-end. A big boost in expansion in terms of capability, and CDMO industry is very agile. We work with a large pharma company to the small biotech. We work with even the virtual biotechs, academia. So our actually theme has been to build the speed, how fast we can accelerate the drug discovery and development process. Key advantages were, as I mentioned, the first to concept to commercial. So start from the very early phase of the target validation to helping in the launching. So some terms called the CRO and the CDMO. So basically now the terms have changed to CRDMO, Contract Research Development Manufacturing Organization, where we support from an end to end. Speed has been our key USP, that how fast we can speed up these services from an end to end services, mostly from a single site. I can share some of the examples. Like I was very fortunate to work with a few CDMOs who supports from an end to end. So your design to the chemistry to molecular biology, in vitro, in vivo, tox, your process development, your non-GMP, GMP manufacturing, regulatory, everything at one site. So what happens? Any large pharma or any of the sponsor, they don't need to work with the multiple organizations. Everything comes at one place, and that saves a lot of time. And again, obviously the cost is obviously it's very much cost efficient, and you are working with the same technology, same what any large pharma is having. So that brings a lot of efficiency as well. One portion I want to add here, that the agility in terms of the CDMOs for a new modalities. We talk about new modalities like yesterday I heard from Dr. Rangarajan, new modality on ADC, oligonucleotides, peptides. CDMOs have been very innovative in this, that how fast we can bring the new technology in place in readiness, so that we can really support this. And again, what to expect, how to bring the nice expectations, set the expectations from each other. So recently we have published, last month came in pharma outsourcing, how to really bring the negotiation with the CDMO and set up on a long-term goal. I'm using some of the data from this year. It's published from an Indian CDMO sector, which mentioned unleashing the tiger. So this white paper has been published from an Boston Consulting Group, as well as the IPSO. IPSO is Innovative Pharmaceutical Service Organization. Major 11 CDMO companies in India have been formed, sit together, and that we need to build an ecosystem. We need to build an influence in the policies, that how we can influence and grow this segment. You see the Indian market share, if you see among globally, is only 2 to 3%. Besides the kind of huge potential we are having, our market is still we are growing, but we are growing in a very healthy speed of 15% CAGR, and we see that still the kind of potential we have, it can within 10 years it can go to close to 300% growth from here. If I compare with the global CDMOs from the US, is obviously if you see the top 5 CDMO in US and top 5 in China, and this is the top 5 in India. Still we are small fractions, and we see that why the word has been used that unleashing the tiger. There are a few key things with India here: talent, availability of talent, capacity to expand. If I compare with someone need to expand the capacity in US and expand the capacity in India, it can be done in 1/3 of the time and 1/10 of the cost. And the talent and new modalities is the one thing which is really fueling the CDMO in terms of growth. Again, that advantage is here, especially this is what has been projected, that how we are looking at in future. You can see in terms of the US FDA approved site in India, we are at number 2. So strong chemistry has been our backbone. Oh, so far, okay. Startup time much faster, and the one thing which has been again going up is how we are expanding the Indian CDMO. It's no longer has been held that everything is in India. We are acquiring the new modalities, new facility all across, and this data has been recently came from all this leading CDMO. There's one portion where I say that how this Indian CDMO sector can really fuel or maybe support the academics, and how we can build a framework. This is the recent data has been published that CDMO can take from end to end, from a starting of this discovery to the end of the commercialization. This is where the key area comes. This is where the academic research institute, where we can have maybe some sort of I would say understanding, setting the expectations from each other, how fast we can bring this from an early stage of the innovation to the preclinical, and how this integrate along with the CDMO to speed up this work. This last slide, again, CDMO has been very underrated in India, but basically it's creating lot of talent, lot of capability, lot of capacity, and again now it has been recognized, and this year has been formed an association how we can influence. Certainly need the pharma CDMO mission. We have been all the policies based on the API generics, which has been our key, but this is the one where we need to really see that, and governing body is certainly being empowered to do that. And again, I always see that if we can set the right expectations from an CDMO industry and the academia, we can certainly get a lot of benefit from each other. Thank you.
Except in your Indian pharma at the present. Now I give to Rashmiji.
Hello. Before I invite the last speaker of the session, I would like to say something. Actually, this is, I think, great opportunity for me to stand before you, because I never thought of sitting besides the very respectable and great personality, medicinal chemist. We used to see sir from very far because he was in medicinal chemistry, and now madam has shown trust in me, and I am sitting besides him. So this is great opportunity for me, and I am really overwhelmed to be here So this is a very wonderful meeting. This should have been organized very early, but now it has started. So this is my humble request to all the organizers of the Alumni Association to please continue such kind of interactions so that we can relive the CDRI life again and again. Thank you. Okay, so our next speaker is Dr. Ravi Thakur from Christ University, Bangalore. Dr. Thakur did his PhD under Dr. D.P. Mishra Endocrinology Division, CDRI. Dr. Thakur is working on the cancer since last 14 years, and his work experience is therapy resistance and tumor stromal crosstalk in pancreatic cancer. So, Dr. Thakur, please start your talk.
Thank you, ma'am. Hello, everyone. So first, I would like to thank CDRI, particularly the organizers, to give me this chance and to inviting me here. And the response has been incredible because I'm mostly a introvert person. I never talk mostly, and it was good to see that most of the people, they still remember me, so that's a good thing. And apart from that, my journey, particularly the PhD journey, started here in 2011 in the old campus. And then I would say, whether I call it lucky or unlucky, that porch fell and then we shifted to the new campus. And the experience has been really wonderful. Means initially for 6 months, the campus was for us only, because when people were shifting, we already came to the hostel, and I think we were the 4 people, those who started the hostel, and then we built up the mess, and then the rest of the things came. And there was a wonderful support from our wardens. They're sitting here, Prabhat sir and Ritu ma'am. So before starting my presentation, it's like whenever I joined, I still remember my PhD interview. I was asked that, okay, what are your favorite areas? I told them 3. First was cancer biology, because I was introduced to cancer a couple of years back when I did my master's dissertation, and I was fascinated by it. Because it's challenging, and from there and then the mission was to identify something or understand it better so that we could find some solutions. We are finding them, but the pace is slow, and the challenge is very, very formidable. And now the the estimates are that out of 10 people by 2040, 4 of them, they will get cancer. Okay, because our lifestyles are changing, things are changing. So a lot of things the lifestyle changes are, I think, the major ones. And that's why I feel that, okay, mission to find novel targets and novel means to treat it are very, very important. So what I'm going to talk about is maybe my journey after my PhD, which I did with Dr. D.P. Mishra. He's sitting here. Thank you, sir, for coming and supporting. And so I joined Dr. Pankaj Singh's lab in University of Nebraska, and he introduced me to the concept of cancer metabolism. I had an idea. I already started a couple of things when I was doing my PhD, but there I saw that, okay, cancer metabolism is something which is a very vast field. And to sum up everything, the importance of metabolism is that we know that, okay, we cannot work until our bellies are not full. Same goes for cancers or the cancer cells. If their metabolism is not functioning properly, or if they are not able to reprogram their metabolism aptly, they will not survive, and that they're doing very efficiently. So we were trying to understand that, okay, how these things happen, how they crosstalk with their local microenvironment, and ultimately how they are affecting the broader physiological context of the body itself. So I finished my PhD thesis, or my PhD presentation, with this slide that I wish cancer got cancer and died. So that was my wish, and it is still my wish. Though it's like I have still found some things which might work, and I'm trying to develop them slightly further so that they ultimately could come out in the market and we could start treating patients. So again, the question always was that how to kill cancer, and the most important is specifically, because we have many means to kill the cells, which also, like, we kill cancer cells, but at the same time we also kill the normal cells, or there are a lot of toxicity that we see. And particularly when we go into the market, chemotherapies are the most common one when we talk about the advanced cancers. And believe me, most of the cancers, those are the prime killers. They are killing us because they are very difficult to detect. Usually, whenever the patient is coming to the clinic, they are already in the third or fourth stage of the disease itself. The only option then and there is chemotherapies, and in certain cases, immunotherapies. But again, immunotherapies, they don't work in most of the cancers. In blood cancers, they are still we had some success, but ultimately whatever we are doing a combination therapy, either combining sometime chemotherapies with immunotherapies for most of the advanced cancers. So it is my hunch, means what I have learned in my 15 years of research journey is that what mostly happens in our body, that we use chemotherapies, in certain cases radiotherapies, and now we are going towards the targeted therapies as well. They kind of kill most of the cancer cells, maybe 90%, 99%, maybe 99.9% also. But there's still some cells, those they're always there, and mostly immune system will be working on those cells and clearing them out. So ultimately, it is a joint effort between whatever we are doing, means from outside, and whatever potential our immune system has. So there was a very inspiring story, which when I shifted from University of Nebraska to University of Oklahoma, one of our veterinarian, she was diagnosed with first time with cancer in 2003, she told, and then she underwent chemotherapy. It returned in 2011. She underwent second chemotherapy. It returned in during, I think, COVID time again. And when I was about to come back, then I heard that, okay, she passed away. So it's like after fighting the cancer 4 times, it always comes back. I'm not saying that, okay, it comes back for everybody, but in most of the cases, we see that, okay, it is very difficult to treat. And that ultimately happens is because one thing is therapy is toxic. Another thing is when cancer is coming repeatedly, it is weakening our body. And there is a term that we call that, okay, body ultimately undergoes a negative metabolic state, which is commonly called as cancer cachexia. And now we are seeing that, okay, even chemotherapies, when they are administered repeatedly, they're also causing cachexia. So ultimately, body is getting weaker. It is going into negative metabolic state. And as I said, that, okay, maybe in the first place we are succeeding, that maybe these chemotherapies or targeted therapies, they might work. They might reduce that cancer to a certain extent. But when the second phase comes, that when our immune system has to work, body is negative metabolic state, immune system, they are no longer functional, or they cannot act, and ultimately it will come back, and then people come to the disease. And that we have seen in multiple cases, that if patients are getting cachectic therapies, they don't work ultimately. So then the idea was that whether we can understand the cancer metabolism, how it is causing these metabolic state, and within cancer itself, whether we can reverse some of it, and at the same time whether we can empower our immune system to survive in those conditions. So that is my prime area of research now, that I'm trying to do, that, okay, apart from finding cancer specific targets and targeting them, whether we can empower the immune system so that whenever it reaches those cancer cells, or whenever it finds those cancer cells, it should be kicking, it should be active, so that we could get a response. So apart from that, the problem doesn't end there. Apart from the negative metabolic state that is present in the body, tumor microenvironment is something which is very, very hostile. So we see that mostly the tumor cells, those are kind of are present in the periphery. They're always surrounded by some immune cells. That means the immune system is always acting on them because they are active. But once we slightly move away from the blood vessels, where nutrients are in scarcity or oxygen is less, and at the same time tumor cells, they undergo the Warburg effect, I think many of we know, so releasing a lot of lactate, thereby creating a completely different kind of microenvironment. So thereby what happens, that now even if the immune cells are reaching that particular point, they are not able to act because the microenvironment is acidic. There is not many nutrients are present at that particular site. So again, the same thing, whether we could find something to enable these cells so that they remain metabolically active, because cancer cells are surviving. They have found those strategy that how to survive in those conditions. Whether we can provide some of these cells with strategies that they could also survive and also act on these cancer cells, thereby ultimately eradicating whatever the residual cells are present. So But before reaching there, my research journey after the postdoc, it started with a very small enzyme, PAGDH, or which takes part in serine metabolic pathway. So when I went, the first problem was given to me that what we at that point we wanted to know that, okay, how cancer cells cope up with different metabolic vulnerabilities. So if there is any scarcity that is present in the tumor microenvironment, how they are surviving. So we did multiple depletions of multiple amino acids, glucose, different other substrate glutamines. And ultimately, what I got particularly related to the glucose deprivation, that okay, whenever we were putting these cells under low glucose conditions, serine, glycine, and threonine metabolic pathways. So serine, glycine, and threonine metabolism related genes, those were getting upregulated. Now it was relatively enigma, but when I deleted the first and rate limiting enzyme of this serine glycine pathway, which is PAGDH, what I found is that now these cells under low glucose condition, they were not able to survive. The question was that, okay, how it is happening? Because if we look at the pathway, so usually serine is being produced from glucose itself. So glucose is converted into glyceraldehyde 3-phosphate, which enters the serine biosynthesis. PAGDH is the first enzyme, and there are two enzymes that are ultimately producing serine. So if glucose is coming from serine itself, serine is coming from glucose itself, how deletion of PAGDH or removal of PAGDH is doing that? Ultimately, we went on to find that it is not a straightforward process. This particular enzyme, which is mostly present in cytoplasm, under this condition will go into the nucleus, and in the nucleus it is performing a slightly different function where it is doing interconversion between 2-hydroxyglutarate and 2-ketoglutarate. So 2-hydroxyglutarate is now kind of a bona fide oncometabolite that is there. It performs epigenetic modification, and these epigenetic modifications ultimately enable these cancer cells to uptake or utilize alternative substrates for their energy needs, and thereby they were surviving even in the low glucose conditions. So ultimately, this was fortunate or unfortunate that this study got scooped in 2020. Another group published the same thing, that PAGDH provides these cancer cells what you call an advantage under low glucose conditions. But it's like we didn't stop there, we moved further. So this particular pathway, ultimately how it goes, that serine produces glycine, and it also provides one carbon unit to nucleotide metabolism. And this particular process is regulated by SHMT1 and SHMT2 enzymes. So, but for these enzymes to work, they require vitamin B6, which is a cofactor. And when we were looking at the tumor microenvironment, we saw that vitamin B6 is present in scarcity. Even in the systemic circulation, we saw the animals, those have tumors, or they are implanted with the tumors, they have low plasma level of vitamin B6. And similarly, we saw in the PDAC patient also that vitamin B6 is low. So thereby we thought that, okay, let's try to see whether we can target this particular pathway, and thereby in blocking the growth of the tumor. We definitely saw that, okay, whenever we are inhibiting either vitamin B6 or this SHMT1 and SHMT2, it reduces the tumor growth, but it poses another problem. There are other cells, particularly in tumor microenvironment, and particularly the NK cells. Those are actually very effective against the tumor cells. They were also utilizing vitamin B6. Okay, so if we inhibit that particular pathway, it's not possible. Another thing is, this is already in scarcity. Tumor cells are utilizing vitamin B6 very aptly, and for other cell types, particularly NK cell and other other immune cells, and the availability of vitamin B6 was very, very low. But fortunately, what we found is that there is a differential utilization of vitamin B6. Tumors were utilizing it to produce their one carbon for one carbon metabolism, but NK cells were utilizing it to deplete to utilize their glycogen stores. So kind of two different vulnerabilities. So what we did, we actually provided these animals, those had tumors with vitamin B6, and what we did, we blocked this pathway SHMT1 and 2 via knockdown, and as well as one inhibitor was discovered, but it was not active in vivo, so we could not do the in vivo experiment. But in vitro, it worked beautifully. Inhibit this particular pathway, thereby regulating the growth of the tumor, and at the same time provide more vitamin B6, which will activate NK cells and thereby providing a better anti tumor properties. So that influenced me, and then I studied NK cells. So, okay, what other vulnerabilities could be there? And there are some that I'm working on to enhance them so that we could devise better therapies. So I'll conclude it because I'm really bad in controlling my time. So I don't want to take much of your time. So that was the major theme. I included some others, but I can skip that. So ultimately, thank you all for listening to me. And the most importantly is that we definitely need a team effort to beat this formidable opponent. And I will definitely look to collaborate with anyone and whosoever is willing to do that. And thank you so much. And yeah, I'm sorry. I always shot beyond my time. I'm not good in controlling.
Okay, that is the sign of excellent researcher.
Very good.
Okay, thank you, Dr. Thakur, for your excellent talk and your approach towards the cancer therapy resistance. We hope that we will have someday the cancer therapy and we will all the patients may survive. Okay, so thank you all for your kind patience. And now we want to conclude the session. We had 3 speakers, wonderful speakers in this session, and they illuminated with their excellent research work, and we definitely got benefited out of it. Thank you all. And I thank my co-chair, Dr. Dinesh Kumar Dikshit sir.
Thanks.
Hi. So I'm sure that you all have enjoyed the very interesting talks from all 3 faculty members. They are the torchbearer of the CDRI and they are doing fantastic in their career. So as a token of appreciation of their effort and the time, we would like to felicitate them, and I request our session chair and co-chair, sir and ma'am, to felicitate the speaker. So in this sequence, I would now call upon Dr. Pankaj Seth, the first speaker of this session today, to please come on the dais.
Chairman is junior to the senior speaker. That's why I asked her. But average age is okay.
Thank you, Pankaj sir. You have provided a very deep insight about how the virus infection and the human brain organoid model serves as a powerful tool for understanding the neurodevelopmental disorders such as microcephaly as well. So now I would call upon Dr. Ashok Jha for a wonderful presentation on the CDMOs and how that CDMO is playing a very important role in accelerating the drug development timeline in India. Thank you, sir. And now I would like to call upon Dr. Ravi Thakur, who just presented, and he has provided a very good insight about how the metabolic vulnerability in the tumor microenvironment play a very important role, and that shapes a new avenue in terms of cancer research and therapy. Thank you, Dr. Thakur. And now the most important event of this session is that facilitation of session chair. So I would like to call upon the chairperson of Alumni Day event, Dr. Amir Nazir and Dr. Vinita Tripathi, to please come on the stage and felicitate our chair and co-chair. Thank you, Dinesh sir and Rashmi ma'am for chairing this session. And now we'll move towards the second session, which is the panel discussion. So we'll take just 1 or 2 minutes to fix the arrangement on the dais. And for the new session coordination, I would request Dr. Shruti to come on the stage and take care of the charge. Thank you.
तो माइक कैसे रखूं? Hello, everyone. Continuing the event, now we have the panel discussion on the topic of fundamental and applied sciences. This discussion is about aligning the academic curriculum with the current demands of industries. So, with immense pleasure, let me introduce the panelists. First and foremost, Dr. Altaf Lal, a celebrated alumnus who has been recognized internationally for his significant contributions in the areas of infectious diseases and public health. Yesterday, you heard his speech about his magnificent journey, both academic and professional, and his roots with CDRI. The second panelist is Dr. Pankaj Seth, Senior Professor from NBRC. He was earlier here on stage for his brilliant talk on human brain organoids. And the third panelist is Dr. Ruchika Yogesh. She is an entrepreneur. Uh, the basic, although the basic training is in the line of organic chemistry, she has recognized herself in the line of medicinal and pharmaceutical sciences. I'll I'll call sir. Ruchika ma'am, with her more than 15 years of industry research experience, she is going to put forward many insights in this discussion. And last but not the least, Dr. Deepak Datta, one of our most distinguished scientists and the current head of Cancer Division. He leads many mission-based projects in CDRI, including Pan India Cancer Program. He has been recognized for his expertise in discovering the therapeutic interventions in the solid tumors, especially the breast cancer. Uh, I take this pleasure to welcome all the panelists, and I request our director ma'am to kindly moderate this discussion. Please welcome on stage.
मैम एक सेकंड मतलब आपका नेम प्लेट लगेगा प्लीज बी सीटेड विल
Where should I sit?
एनीवेयर
There?
डोंट वरी विल मैनेज दैट पार्ट
Can you hear me? Good morning, everyone, and welcome to this panel discussion. We did hear about a topic from the from Shruti, but I intend for this to be more of a freewheeling conversation. So I have not really planned questions along the lines of the topic, but we're going to have a more of a conversation here. I hope you don't mind. Two of our panelists have already spoken earlier, either today or yesterday. Deepak is a current faculty here, and Ruchika, right? Dr. Ruchika is an entrepreneur who's also whom we haven't heard from, but as this conversation progresses, we'll get to hear about her journey as well. So, Dr. Altaf, I'm going to start with you. You've had a really interesting career. After leaving CDRI, you've worn many hats. You've been in industry, you've been on the regulatory side, you've been involved in policy, you've been involved in drug development from many different angles. So we'd like to understand, or I think for the students here, it's interesting to learn about these types of career changes and career choices, because there is a certain assumption that you start off in academia, you stay in academia, or you start off in industry and you stay in industry, and not very often do you find people going back and forth or changing altogether and doing something different. Public health has been a theme that resonates with all stages of your career, but the roles you've played have been very different. Perhaps very briefly, you can talk about why you made these changes, what was the thinking behind it, and do any of these roles that you played have to do with some of the experiences you had at CDRI, which exposed you to perhaps the world of drug development?
Thank you so much. Someone asked me something very related a few days ago, and my answer was that I'm a vacuum cleaner. CDRI laid the foundation to work hard, to read, and to ask questions. And then came NIH, which was completely basic sciences. And Prant Talwar had to be at NIH as a Fogarty Fellow one day. And he said, "Ye kya kar rahe ho? Actin or myosin pe kuch aisa kaam karo jo desh ke liye acha ho." And that sunk in. So listen to those who know more. And I've been a constant listener. And I've listened to everything that was said over the last 24 hours. While we were talking, I was creating partnerships. Man to my left has a superb collaborator in making. She's at University of San Francisco, hopefully coming back to India. So NIH provided the basis to me that working on mice and monkeys is not the right thing always. You got to work on human side of the diseases. So that came the CDC chapter, a very vibrant 30 years. And that exposed me that you can do science, but at the end of the day, the science has to be converted into policies that would be realistic and timely. And I was asked to come to India for 2 years to serve as a health attaché, build US-India relationships. 2 years became 7 years. And I had to get fired to go back. The reason was simple, that once you've done something for X number of years, I think you've done everything in the X number of years in that little space. And then came the private space, then the regulatory space. The regulatory space became very important because what we make, how we make is important, and how do we convince the patient that what we make is one, affordable and safe and effective. It just was a transition from one to another that was seamless. And I haven't yet talked about the charitable space I operate, but that's a conversation for some other day. So my advice to you will be, for those of you who are younger version of me and younger version of the Baron Director, just read, extensively read, and make sure that you're asking questions, what next? And that'll come in your life, and you'll, inshallah, be very successful.
Thank you. So read, listen, and then take action. Right? Okay, so that's a really different or a proactive approach to life, saying that, okay, you take a certain path, you remain open to change, you remain open to directional shifts, and as you evolve as a person and as your understanding of the world changes, adapt and find the role that fits you as who you are best, right? So I'll ask you a different kind of question, Pankaj. When you think back to your CDRI experience, would you like to talk about one high point and one low point? And then do you also want to talk about how you felt when you started the position that you took on, right after you left CDRI? Did you feel well prepared?
Yes. So, as I think all of us have been saying repeatedly, this foundation which was laid in CDRI was rock solid. And that prepared me, at least, for, and I think most of us, to do anything which we could. I started working on a blood-brain barrier model as a summer trainee in CDRI.
Oh.
So I started very early, as I was telling in my talk. Then I started working on thrombosis with under Dr. Madhu Dixit's guidance, and that laid the foundation. And I think that's my high point, is that when I was in that lab and I started working with them, and it just brick by brick, it laid the foundation to be on the high point. And I was also very terrified when I was being asked to give a talk to start with. And I think in Department of Pharmacology, or maybe in CDRI, I'm talking about 1991, 92, we were the first lab which had a JC, Journal Club. And we didn't have a PowerPoint presentation, so we had a blackboard and a chalk of 2 colors only. So we used to prepare our slides or our presentation, whatever we wanted to talk next day, day before. So, and I would not sleep that night. But gradually, brick by brick, as I said, Dr. Madhu Dixit made sure that I don't squeeze away, so I can't fall sick. And then I started enjoying it, and I started to read, as Dr. Lal mentioned, and that's made a foundation for me to learn more. And to ask questions, I was always pushed at home. So here we had Dr. Madhu Dixit. Back home, I had Dr. P.K. Seth, my father, and there was no escape. So gradually that built into me to ask questions, and be inquisitive. And in many of my slides, which I now start give more of the motivation talks also, I put inquisitiveness is the paramount. So these were the high points. I was really well prepared. I was good at thrombosis. I was good at IV injections. And when I was in my summer training, I was going home and telling, I can do any IV injections. I can do in rat, mice, anything. My dad said, you're not going to be a technician. What is the basis? Why are you doing that? So that again helped me to do better. And when I went for my first postdoc, it was in a Department of Defense lab. And there we started asking about ischemia reperfusion, which was very similar to what I was doing in thrombosis. And that helped me do well in that lab. We published a series of papers. And then I switched over from USUHS in Bethesda, one road, and went into NIH. And that my infectious disease journey started. So that is my high points. I think my only low point once was I wasn't sure whether I'll be able to pull through this. But again, as I said, my dad was like Amitabh Bachchan of toxicology, neurotoxicology. And I always tried to do well and to keep up to his expectations. And he really encouraged me to do that. I don't remember too many of my low points because I was in a very good environment. I had Dr. Pandey, who was in charge of the instrument section, and Madam was again in charge. So we had all the new equipments which would come to pharmacology. And the day the demonstrations will be there, I'll make sure that I'm there, so I know also how to run it. So we had CDRI, a very strong foundation. I was good at instrumentation, bioassays, and that helped me prepare to become a good scientist.
Wonderful. Thank you, Pankaj, for sharing your experiences. Clearly, you remember, your memories of that period are one of great learning and, one of being well supported both in the lab and outside. That allowed you to develop as a scientist and believe in yourself, right? Believe that you could do it. Perhaps, in hindsight, we forget the challenges that we had to overcome, because no PhD experience is without its challenges and without setbacks, without realizing that perhaps one had chased the wrong thing or had taken a risk, pursued a particular direction and found out that that wasn't particularly the right direction. And we see that frequently in drug discovery because there's no way to know ahead of time how molecules will behave, what the properties will be in vivo, what kind of toxicity, what kind of safety we are going to observe. So, Deepak, as someone who is balanced between basic research and translational work, and because I've watched you so closely over the last 3 years, I know that you have equal levels of passion on both sides. And I don't know how you do that, but that's really to your credit. When you're chasing basic research, there's one set of risks. When you're chasing translational questions, there is another set of risks. So when you're with translational research, where a lot depends on chemistry, biology, pharmacokinetics, and so on and so forth, how do you deal with the challenges that arise with this kind of uncertainty? How do you hedge your bets so that when something fails, you can pick up something else?
First of all, I am greatly honored, ma'am. Thank you very much for this opportunity, CDRI Alumni Group. So definitely, I think I should say CDRI is possibly the only academic institution in the country where you get an opportunity to balance both of them. So in that regard, we are very, very fortunate. Here, every faculty, if somebody wishes, will get an opportunity to balance translational research and basic research, although they are not definitely separate. I truly believe it. So what Dr. Lal said, very important questions, like, keep asking the right questions from the very beginning. Although it's very challenging in the beginning when we joined and started, and not only from abroad, when you join here, there are different challenges like purchase procedure and all these formalities you have to maintain. Jam recently in last 3, 4, 5 years.
Make in
So
India.
your life got miserable and incredibly challenging. But in the very beginning, what you asked, you may see the fruit in 10 years later, I would say. So that hindsight of the first 5 years of your joining, it's what you reap, what you sow in the later part of the year, in like 10, 12 years, because here the pace is very slow, I would say. It's not like abroad, every 2, 3 years you publish a big papers and move on. Here you're like, if you ask the right questions in 10 years back, you may see right now the fruits, what we have been seeing for last 3, 4 years, man. And the most importantly, what you asked, how do we balance like these two part. So when we present in different podiums, I always first slide divided in two bilateral symmetry, just like human being, the slides. One part 50, like from the very beginning we try to say 50% drug discovery, 50% fundamental research. And from the long run, I know very well, if you ask the right questions in the fundamental research, those proteins and those targets, and those proteins could be your next generation targets. Exactly what happened, like what we published in a couple of years back, right now is our target for doing a lot of translational research, just like easy is to what we have been working. And so next onwards it would be very easy, but in the beginning it was very challenging. And actually it's working in both ways. You are de-risking yourself, your student's career also. Not only, it's not risk, actually you are de-risking, because any one of the time your basic research fails, you can always complement with some molecule driven work, and your molecule is always risky, you never know when it will get dropped. So your basic research will support your fellow's PhD career. So actually it's a blessings, not that so much difficult. Difficult part is when you don't have funding, you have huge purchase procedure, again I am mentioning, and you are struggling with 2 lakhs of money in the whole year, and you will adjust yourself accordingly. And that time you learn a lot of very critical things which you have not learned before also. So just like I am giving an example, at a time when our DST grant was spending money, we had a very short of money. That time the whole lab converted from siRNA mediated knockdown to all lentiviral system knockdown or CRISPR based knockdown. So it was, although very challenging, but it blessed us to move forward in the modern technologies, man. So it is actually blessings to balance it equally in both translation and fundamental research. Thank you.
Thank you, Deepak. Very insightful, and I think very relevant for our for the CDRI community and our alums to say that these are not very these are translational research and fundamental basic research. These are closely intertwined, and if you can use both well, then you're actually de-risking on both sides. That's an interesting perspective. Most people tend to gravitate towards one or the other, believing that the other side is too difficult. But your example shows us that it's actually possible to do both, and essentially one feeds off the other. This is the core principle of CDRI anyway, that there is basic fundamental research that's going on, and some percentage of that work will convert or translate into product ideas which can be pursued differently. Yeah. So I think for students as well, the learning is that if you are on both sides, you're actually getting the best of both worlds, and you will that you will distinguish yourself from students coming out of other institutions where the possibility of doing both things is simply not there. So, Dr. Ruchika, I would like to ask you some questions about, your career choice. I would say that most students who finish their PhDs at CDRI take an academic path, which means to go do a postdoc, and then typically after a postdoc to do an academic job. We have students who go into industry as well, and many students out of pharmacokinetics, toxicology, and medicinal chemistry divisions tend to do that. But it's still I would still say that it's a minority. I don't know the numbers, maybe less than 30%, something like that. And very, very few become entrepreneurs, right? It's still an exception. Even though in our country today, we talk greatly about entrepreneurial initiatives and efforts and government support for it, finding somebody who is willing to leave the path well trodden and choose to risk everything and be an entrepreneur, that's still an exception. So would you like to talk about the your decision and why you made the decision and how you're liking it?
Thank you, ma'am. First of all, I'm still in the awe of sharing the dais with you all the stalwarts. Thank you for providing the opportunity and asking me this question. So the first thing is that being entrepreneur is not something which is very new or people can't do it. Everybody has got ideas. It's just that you need to take that stride. You need to have that courage, basically. So just to, if I'm allowed to share my journey, after masters, I did my masters from Banaras Hindu University, and just for knowledge, I was the gold medalist there. I topped the university at that time in all master courses. And after that, I just wanted to go to some work somewhere and join industry. Basically, I joined Ranbaxy Research Laboratories at that time. There, I realized that without PhD, I'm not going to go anywhere. I'm just an MSc, right? So, as Dr. Dikshit, Madhu ma'am was saying yesterday, in your career, several people come who would tell you, or help you somewhere at some point, which actually shapes your career in later years. No, so one of my seniors, she said that you're being a topper. Why aren't you going for JRF? I mean, CSIR-JRF exams. So I was like, yeah, okay. And then I went for CSIR-JRF exams. And while being in Ranbaxy, I qualified. And then I got the JRF ship. And I came here because I was already introduced to pharma, that environment. I was already introduced. I chose to come to CDRI. I met Dr. Saxena. And what CDRI gives you is, the first thing that CDRI gives you is purpose, right? Why are you doing PhD has a purpose. So that was the first spark wherein you get that you have a purpose for doing your PhD to live your life. That is drug discovery. Even if you don't discover, at least you do something around it, right? So my entrepreneurship journey would be starting from that point wherein you are joining CDRI with a purpose of drug discovery, right? And then, of course, as Dr. Altaf was saying, your career is not, bound by things you've started with. You can always, keep on changing if you have that, I won't say metal, but then, yes, you have that resilience, basically, that perseverance, that resilience. I'm an example wherein I started off with a job as a chemist in Ranbaxy Research Laboratories. I came here, I completed PhD, and then I went back to Ranbaxy. Then I left Ranbaxy for my children, because after all, everybody wants family, and I was married. I had 2 children. I left. Around during those 4 and 5 years of my break, I trained myself again as a copy editor and a writer, because that was the profession where I could have bridged between my studies, whatever I have done in the past, and earning money, right? And then I came back. I joined Curious Journal of Medical Sciences as a copy editor, and I'm if anybody wants publications, reach out to me. Maybe I can help. And then I started 2 startups. One was because I was highly mesmerized with the power of AI. So I started one in the field of AI, and then Mattis Labs, wherein we Mattis Lab was again identifying that what are the problems that you can solve. Coming back to your question, ma'am, here, that being an entrepreneur, you need to identify a question, a thing that could provide solutions to a lot of people's problem. So that is how you go into entrepreneurship. And if you're doing that, in that journey, you learn a lot. You learn, going and joining an institution would give you all the things that are already set up. You just have to go in and make your place. But then being an entrepreneur is right from the scratch. You do everything. You do you just set up, you go into all legalities, you set up a system, you choose people, you make, so right from choosing your co-founder to choosing your team, you're doing everything, and I mean, just to live up to your dreams. So yeah, entrepreneurship is all about finding a problem that society needs a solution for, and how you can contribute to that.
Great, thank you. So you made some really, really interesting and important points. One is your story is a story of someone who came back from a break, and there's a question that many professional women face. Different people make different choices. There's no right choice and there's no wrong choice, but we all struggle with that choice. If you take a break, you feel like your career is going bust. If you don't take a break, you constantly worry that somehow you're not being a good enough mother. And so there is that double whammy that goes, it's a constant thread. So you made a choice to leave, and then when you decided to come back, you reinvented yourself. So again, the power of believing that you can do something different, chasing that dream, and then you decided to start your own company. And don't forget, if you're running your own company, you're responsible for payroll, right? Everybody's salary at the end of that month is on you. So that's a very, very different kind of responsibility, right?
Not that month, actually. You need to prepare for at least
a few
6 months and ahead.
Yeah.
You need to have that balance.
What I meant is that
because
amongst all, over and above all the different roles that you play, it's also the payroll that is a big responsibility. Again, so you feel compelled by some purpose, and that drives you, isn't it? Because that purpose makes the challenge worthwhile. So seeking or finding your purpose in life, that's very, very important. You got to find something that really moves you, that really pushes you, because there will be times when you feel like stopping and giving up. There are times when you say, I just cannot do it. But then the next day you get up and you say, I'll do it, because you are moved by something bigger than yourself. And you talked about this, Deepak talked about it. The ultimate purpose is what can we do for society, whether we are developing drugs and changing somebody's life, or we are making life simpler or easier for somebody by offering a service. So that is the ultimate motivation that makes the challenges worthwhile. So I'll come back to. We were running late.
The clock is not moving, so we have all the time.
We've been given a bonus. Very good. We can keep talking, forever. So you have to really stop us. Okay, we'll do one round and then we'll see if we have time for one more. What advice would you give to students pursuing PhDs these days? And each of you answer that question.
I think my first advice will be to the mentors, to the thesis advisors, to the faculty. We grew up at times when the science traveled by slow mail and we read the journal. We were less competitive. You, the thesis advisors, have to become students at the same time that you are thesis advisors. Mm-hmm. You have to live the lives of your students. Encourage them. If you can, have them refer to you by your first name. Amir, Madhu, Deepak. You're going to prepare them for the life, a better life, a competitive life, an aggressive life. Aggressive not in the sense of fighting, but a life where you are co-equal.
Right.
So that's a very important transition. CDRI की तारीफ करना लाजमी है. 30 साल बाद हम ये तारीफ कर सकेंगे कि नहीं, उसकी तैयारी करें आप. Right? How do you prepare for that? And I would have loved all the students in the back row to come up front. Those who are sitting in the back, you have told yourself they're backbenchers. You're not backbenchers. I think in the next 2 minutes, if you can all come at the front, you will see the room will change. It'll transform. So faculty is very critical. And now we live in a day and time where whatever is done in Seattle is known in Singapore, and whatever is done in Singapore is known in Lucknow. So take advantage of that.
Thank you. Pankaj, would you like to comment on that? Question?
I think he's put it very rightly. We need to worry about, or I should not say worry, but we should plan about that. How do we take the graph, continue to take the graph of CDRI like this? And for that, we need to work yesterday, actually, not today. I think that for the students, I would say there are no shortcuts. Okay. First thing is always go for high-hanging fruits. Don't go for low-hanging fruits. And as Dr. Lal put it very rightly, we are in a very, very competitive things. If I can make organoids in NBRC, I don't know how many people have already done it elsewhere. And everything is very, very easy to do, easy to read. So find out ways how you can get to the knowledge before anybody else does. And then don't start from there. You start from somewhere here, because as Dr. Deepak said, our systems are comparatively a little slower. So by the time you'll get to that, and in US, and NIH, we used to order something and it used to come in next day. Mm-hmm. It doesn't happen here. So when I came here, I made myself very clear about it that, forget about what you had privileges in US, and you see how you're going to succeed in this environment. So to the mentors who have recently returned from US or anywhere from the world, हाँ, उसको वहीं छोड़ दीजिए और ये देखिए कि यहाँ के सिस्टम में आप कैसे बेस्ट सर्वाइव कर सकते हैं. So I'll go back and forth with mentors and students. So yes, Deepak, I know I also struggle with GEM. So what I did was I have delegated things in my lab. So first of all, I did a course which was offered to me from DST. There was a course which was conducted by Management Development Institute in Gurgaon, which is something like as an IAM. It's for executive MBAs. I did not do an MBA like Dr. Lal, but I did that course, which was very helpful. It was on science policies and management, managing of personnel. So I identify in my lab, and whoever is there close by, I try to understand who's good at what. So every time when we get a batch of students, I figure out who's the computer geek. So I assign few things to him or her. And then in my technical team, I have a technician and a lab assistant. I figured out this guy cannot do much, but is very good at internet and everything. So I said, तुमको जिम का इंचार्ज बना दिया, सबको इंचार्ज बना दो किसी चीज का. तो इंचार्ज बनाने से क्या होता है, उनकी रिस्पॉन्सिबिलिटी बढ़ जाती है और आपकी खत्म हो जाती है. और ये मेरे डायरेक्टर्स मेरे साथ बहुत अच्छा करते रहे. मुझे अब रियलाइज हुआ कि क्यों ना एवरी टाइम आई विल कम होम एंड आई टेल माय वाइफ हु इज सिटिंग देयर, who has sacrificed her career for our kids who are doing well. But मैं रोज किसी चीज का इंचार्ज बन के घर आता था बड़ा खुश, और 3-4 दिन बाद समझ में आता था कि कहाँ फंस गए. So the mentors, please keep a balance that while you are relevant for your institute and you help in institution building, you play a role which has been given, but be careful to draw a boundary and learn to say a little bit of no. But for students, as I said, हम लोग अपने रिसर्च प्रोजेक्ट जब भी डिस्कस करने शुरू करते हैं, कुछ सोचते हैं, सबसे पहले हम एनआईएच की वो जो रिपोर्ट होती है जहाँ पे all NIH funded grants are listed with their objectives and an abstract. We do literature search, but we also go and search there ki what has already been funded. So if something is funded and that idea is funded, by the time you will start working, they will have a paper. So your scooping will be a little minimal. I'm not saying it's a guarantee. So look for your ideas, go to report and check whether, and often you'll find that what you were thinking is already funded somewhere by NIH. So, and then, like I did, I started with human neural stem cells. We are the only one, Director Ma'am, even in India, in today, who has the primary cultures of human fetal brain-derived neural stem cells. It's fortunate and unfortunate in the country that we are the only one at NBRC. Why we are only one? Because it was hard. It started, first 2 years after I came back and joined as a scientist from my postdoc from NIH, went into ethical clearance and getting human fetal tissue.
Right.
And I'll just take a minute.
Got stuck in that.
Yes, but I never got into the, and thanks to my dad, he always told me, don't get frustrated. Every evening I'll call him and tell him, ye hua, wo hua, ye nahi, wo nahi. But then he said, he would hear me out and then tell me one thing: don't give up, don't give up. So no giving up. There's no chance of giving. If you get tired of something, if you get frustrated, do something else other than in academia. Be prepared for it. This for students as well as for the mentors, and more for the mentors, because student will come to you and look at your face and then decide whether to ask that question, whether to ask that permission, whether to talk to you or not. Often student, because we are sitting in those glass huge window cabins, and they will say, रहने दो यार, आज boss का mood ठीक नहीं लगा. So no matter how difficult it is for a day for me, I had done one thing for myself. I had never turned away any student. In fact, I've assigned days for everybody: Monday, Tuesday, Wednesday, Thursday, Friday, Saturday, Sunday also, but half days. I don't go anymore on Saturday, but I'm available 24/7 to them. And that I learned from a college principal in Gurgaon. So once I went to meet, I give lot of these scientific talks nowadays, and I saw the board बाहर principal का लगा था, government college का. Principal में मिलने का समय विद्यार्थियों के लिए कभी भी. So I was so impressed. I brought a picture and I showed to my sons and my wife and to everybody. I used to use it in my slide also. So be available for your students, understand the problem, and go for a high hanging fruit. So fetal tissue का 1 minute में बताता हूँ मैं. I used to first I got over कैसे करना है, informed consent के वो सब कुछ मुझे अच्छे से समझ में आ गया. I started writing good projects. मेरे proposals कन्विंस करके एथिकल अप्रूवल्स हो जाते थे. बट द प्रॉब्लम स्टार्टेड, मैं गया फीटल टिश्यू, अबॉर्शन का फीटल टिश्यू लेना है, पहले आपको गवर्नमेंट हॉस्पिटल से, तो ठीक है, लाइजनिंग हो गई, ऑल फीमेल डॉक्टर्स, सब कुछ ठीक है. उनका फोन आता कि डॉक्टर साहब आ जाओ, एक अबॉर्शन है, 4 है अबॉर्शन है, आ जाना सुबह से. आप पहुंच गए डॉक्टर साहब अपना डब्बा लेके नॉर्मल सलाइन का. अब मुझे पहला है चैलेंज कि मुझे अनइन्फेक्टेड लेके आना है, मतलब नॉर्मल फीटल टिश्यू लाना है, बट स्टेराइल कंडीशन में गुड़गांव हॉस्पिटल से मानेसर आना है, विच इज अ 20 किलोमीटर ड्राइव. टिश्यू पहुंच गया इन्फॉर्म कंसेंट पे, पहले पूछा मदर से, नहीं जी हम नहीं देंगे अपना बच्चा। ठीक है, आई अंडरस्टैंड। 10 मदर से पूछा, एक रेडी हो गई, मैं जी हस्बैंड से पूछ के बताऊंगी। हस्बैंड भी रेडी हो गया, उनको बताया क्यों क्यों करेंगे, क्या करेंगे। फिर रुक जाओ जी, अपनी सासू माँ से पूछ के आएंगे। तो दिस कंटिन्यूड, बट आई नेवर गिव अप। आई से ठीक है, कोई बात नहीं, हम 20 लोगों से बात करेंगे। एंड आई यूज्ड टू स्टेशन देयर, कैंप देयर, समोसा चाय जो भी है वहीं पे बैठे रहते थे, लंच नहीं कर रहे थे। बट वो पहले के 1 महीने के बाद मुझे समझ में आया कि कैसे क्या करना है, एंड आई सक्सीडेड। आई गॉट द टिश्यूज, आई स्टार्टेड द वर्क, आई स्टार्टेड गेटिंग फंडिंग। और मेरा पेपर एक्सेप्ट हो, रिजेक्ट हो, रिवाइज हो, एक लाइन कंसिस्टेंटली आई ऑलवेज गॉट ऑन द रिव्यूज फ्रॉम द एक्सपर्ट्स. This is a very challenging primary culture system, which is the strength of this book. So always identify how you have to be different from the routine, and that will happen only the harder way. So there are no shortcuts in research. You can use as much as AI you want to, but you will have to use your brain and knowledge and reading and experience. Please follow that. No shortcuts.
Okay. So actually, I'm going to change the question. I think the one question that I really do want to ask is from you alumni, and also from those in the audience. We ultimately want to be recognized as a global center for drug discovery. We need to envision the future and raise the bar for ourselves, rise to a higher level, become, we have a certain national reputation. We have to go beyond that. We have to do more. We have to become that global center. If we agree that that is our goal long term, what in your view is could be ways in which alumni could help CDRI with this goal?
Can I?
Yeah, please.
I think that's a commendable target. The term global, which means you're above national, not that national goal isn't good. It truly requires a group of people sitting around the table, defining what our deficits, not what is good about us,
yes,
but what we do not have.
Right.
I would not look at the past. I would look at what are the 5 conditions or 6 conditions, infectious or chronic, that we can take a lead in, that the rest of the world, for a variety of reasons, economic or other, isn't paying attention to. We identified those 6 or a number that the leadership decides. And then there is a pathway. We're going to have quarterly, like a business. CDRI then has to be thinking through an entrepreneur's mind. Each PI has a quarterly, bi-quarterly internal assessments. Kya hum kar rahe hain? Hamare paas systems hain. That comes to, again, the Lucknow and New Delhi leadership. Enable them with the processes and the systems. A chemical order comes in a week, not 2 months. A travel that is needed to happen in a week is approved in a week. An accountability framework. One thing entrepreneurs do is they surround themselves with accountability framework. Agar koi cheez nahi chal rahi hai, we'll just figure out kyun nahi chal rahi hai. Nahi chal rahi hai, fir bhi kar lenge. You'll just find a solution. You will find a solution. For those of you who were born after 80, and I think many of you are born after 80, when thermocycler, the PCR, right, came into existence, NIH, which was heavily funded, we had 3 water baths, 90 degrees, 70 degrees, 34 degrees. We kept moving and burning our fingers at the same time. We did not stop. That's some of the best science came from those 3 water baths. So you can have those 3 water bats here, water bats of engine, water bats of what will make it global, but it needs a business thinking. It needs a business creativity mixed with science.
And what do you see as the role of alumni?
I think they should be at your disposal. You should, without any hesitation, say, well, we're stuck here. A group of 8 people who've traveled the distance, people who have failed, as well as succeeded. You're going to create something in a product line, which is either a diagnostics or a chemical entity or phyto entity. I don't see much biologic. Maybe there is some biologic,
not much.
Not biologic. So the definitions are very clear. The chemical entities, and also figure out, agar मैं कुछ ये कर रहा हूँ, agar institute कुछ मैं becomes institute now, right?
Yeah.
Everyone is an institute. If I'm proposing this diagnostic test, what are the differentiators?
Right.
Is the market saturated? Is there a market need? Agar nahi hai, it's an academic exercise. I'm not going to put my money into there, money and time.
Right.
But if there is a chemical entity that has a differentiator, think like an industry. And you would find people who'll be willing to spend time and efforts and resources to be available to you electronically or in person. And if you need me to vacuum those individuals for you, I'll be happy to vacuum those individuals for you.
Great. So it's something like use the alumni as in getting unstuck from whatever.
Yeah. And alumni should know that they're coming here as mazdoors. You're not coming with attitude. Ki mujhe aata hai. You got to enable the system. We're shown the time is up, and that's exactly what happens in product development. Time is up because someone has put in the market. So isse pehle koi aur market mein dalta hai, you should have a claim on it. Yeah. This is a larger conversation, but I think you're on the right track.
Great, thank you. Pankaj, very quickly, maybe.
Add on to that, I think it's time, CDRI has its people everywhere.
Right.
You look at the front row. Dr. Khan, he just retired from ICMR. And I'm sure many more are there already in ICMR.
Yeah.
We have people in regulatory affairs. We have strength who are, like Dr. Ranjana, who was just sitting here, is an expert in regulatory affairs. She knows that system. And similarly, the basic scientists, clinicians, everything is everywhere. We need to identify among those alumni who's good at what,
strengths,
their strengths, and then figure out and let the current students and PIs know that, if you need help in this area, you go there.
Right.
And we form some kind of a system where, people can just, like in LinkedIn, you put in your request, I'm stuck here. Can you help me? Or can you guide me or doing this? And then alumni has to give back. This is time to give back to the community. And we're all willing to do that.
That's wonderful. So, sort of like a directory almost, and a way to, go and ask your question. And, that it's not like you're not throwing the question out into the ether. You're actually talking to a very committed group of people.
To Rahul, and we figured out how to work on a Zika virus, the dengue antibody, and all that.
Yeah.
An organized system.
Great. Would you like to answer that question, Ruchika?
Yes, ma'am. I was just so as Dr. said that's the thing I was going to say, that while working in Ranbaxy, I was a process chemist, but we had to work with all the departments, right? Analytical, DRA, and all of the other departments. So there, I understood that if you're talking about drug discovery, or actually you're going to do something meaningful in this field, you have to have knowledge of all the things. You need to know that how your molecule is going to behave when it is exposed to all toxicological studies. So you have to have that knowledge. So rather than working in silos, what alumni can do, that if you are choosing something as your problem, as a medicinal chemist, if you're choosing some problem for you, just go and make a group of other people working in other disciplines, and actually make out the best of it. And as we say in our entrepreneurial world, that make fast, fail fast, learn fast, and come back fast. So that's like, instead of chasing something, spending a lot of time that we have done, just do fast, fail fast, learn fast, come back fast. So
Great. Thank you. Yeah. So, set criteria for progression, set criteria for making decisions. When you get to those decision-making points, don't shift the goalposts, don't shift the criteria, follow those, and then, use alumni as needed. And Deepak, question to you. You're fully aware of our system, the good, the bad, the ugly, the gaps, everything that, this is our lived experience. Where do you think alumni can contribute most?
I think just echoing with what Dr. Lal said, also Dr. Pankaj said, like LinkedIn and Zoom का ज़माना में बहुत आसान है interact करना. So I would say beyond interacting in online platform, also we can do a couple of things so that we can make it very in regular ways. Say for example, we do organize a lot of conferences all through year. So now we have conceptualized the whole alumni platform. We know wherever it is some alumni sitting, where, especially in the abroad, especially in the industry, and who do they are doing it, in which positions. We can track up them when they are coming to the country. We can always catch hold of them, come and visit CDRI, especially very close interactions with particular disease biology areas as a team, and interact with them in person also, so
Right.
that we could get the real vibes of what outside is going on,
Right.
especially from industry point of view. And also we organize again conferences, some sections for the whole alumni talk. By that way we can interact all through year. What we right now conceptualize, maybe we won't see that effect in immediately by through online, definitely, but in 5 years time frame, if we could practice this in 3-4 conferences in that way, definitely we'll crystallize something very solid. What Dr. Lal is saying, we could get what right now it's going in the frontiers of the world, and what targets need to be chased, and what kind of frontiers basic science we could do from their ideas.
Okay.
So that could be our next course of action, maybe after conceptualization.
Great. Yeah. I mean, from my point of view, I'd love to see more collaborations with alumni in whatever way possible, but really in more tangible ways than being there to provide advice and acting as a sounding board. What research collaborations, projects can we work on jointly? Can we identify a few and start them? Because if we have success with one or two, then it gives us confidence to expand that model. Can we get help on business development? Can we get help on licensing? Can we get help on, can we get your inputs into how we can begin to ask the high-hanging fruit? In the Indian system, because of all the constraints and because of all the difficulties with working within it, you get into a mindset that, I got to get something done. So let me do something which is a little bit simpler. But in doing that and taking that approach, we fail to raise the overall standards. So there are some real challenges and there are some real difficulties, and not all of it can be changed because it's a publicly funded institution and we are part of a bigger network. But within those, accepting those constraints as a given, it would be great for our alumni to really help us fuel research. This could even mean creating avenues for research projects and for funding, for travel and things like that, which could help our scientists and our students, particularly, travel outside the country, participate in conferences, and so on and so forth. So I think there are a number of different things that could be done with alumni, and I'm hoping that with the association crystallizing, there'll be some definite pathways in which CDRI can formally interact with alums. I would have loved to open the floor to questions from the audience, but I know Amir's been looking at me steadily, giving me the dirty look. So we're gonna stop here. I want to thank you all for really a very, very wonderful conversation, and for sharing your, your candid feelings and thoughts. It makes us feel very, very, very connected. It gives us that sense of comfort in reaching out and asking when we need something. By the same token, I'll say also, if we can help you in some way, we will always be there as your alma mater, supporting your endeavors and taking pride in what you achieve. And if there are things that we could do for you, we would, and we stand by that. Thank you very much, and thanks to the audience.
Thank you, dignitaries. That was indeed very insightful discussion. If I have absorbed and understood properly, the takeaway is basically the synergy. So the synergy of producing innovative and, as Pankaj sir said, adaptable ideas, while we hold the hand of the skill gap which can satisfy the demands of modern workforce. Am I okay? So and that concludes this panel discussion, and I hope you all appreciated the perspectives put together in this discussion. And I would like to request our director to kindly felicitate our esteemed panelists. First and foremost, I invite Dr. Altaf sir to please come forward and accept our gesture of appreciation. Dr. Pankaj, sir, please come forward. Dr. Ruchika Yogesh, ma'am, please come. And our sincere thanks for accepting this in the last minute. Thank you so much. Dr. Deepak Datta, please give a big round of applause, people, please. Thank you, ma'am. May I now invite Dr. Amir Nazir, chairperson, and Dr. Vinita, ma'am, co-chair, to please felicitate our moderator, Dr. Radha Rangarajan. Please, ma'am. It's okay. Thank you. Thank you so much. Now I humbly invite you all for a cup of tea. And after this, there will be area-wise discussion. The life science team will be having the interaction in the hall.
Plan to have it here only.
Both.
Yeah. So 10-minute tea. Announce 10-minute tea and assembly here.
Okay. So I request you all to please assemble after 10 minutes of quick tea break. Thank you.
So 12:20, please be back. So we'll have a quick 10-minute tea and then we will do. Yeah. So we request all the students to be back because we'll have this interaction. Now we have combined both life science and chemical science teams and we will assemble here only. So 12:20, please be back. So what we'll
Very good afternoon, everybody, and welcome back to the last session before formal closing ceremony of the event. And this is going to be a student interaction session with some of our selected distinguished alumni. I'm sure students are going to learn a lot from this session. And for this session, I would like to invite first Dr. P. Srinivas. Dr. P. Srinivas did PhD with Dr. Kalpana Bhandari in Medicinal Chemistry Division in 2008. And after working extensively in pharma companies in India as well as in Germany, he started his own CRO NS lab in Hyderabad in 2015. Dr. Srinivas. Our another alumni today is Dr. Chitra Gupta. Yeah, sir, please come. Dr. Gupta did her PhD from Biopolymer Division with Dr. Shyam Singh in 2001, and she is currently working with GlaxoSmithKline Pharmaceuticals as Head of Government Affairs and Policy Advocacy. Dr. Gupta. Another panelist for today's session is Dr. Manish Dikshit. Dr. Dikshit did PhD with Dr. Atul Goyal in Medicinal Chemistry Division in 2007. He did postdocs, couple of postdocs at University of Arizona and then NIH Cancer Research Center in USA. In 2013, he joined CDRI Lucknow, where he is currently working in the field of nuclear medicine. Our fourth alumni for the day is Dr. Tushar Srivastava. Dr. Srivastava did his PhD with Dr. S.B. Katti in Biopolymer Division in year 1999. He worked in almost all reputed pharma companies in India, Ranbaxy, which is Sun Pharma currently, Lupin, Mylan, Unimark Remedies, and in 2016 he founded OMSA Pharma, which is headquartered in Mumbai, and research facility, R&D facility at Ahmedabad. Our fifth and last panelist for today's discussion is Dr. Anjana Seth. Dr. Seth did PhD with Dr. Madhu Dikshit in 1995. She did postdocs at University of Chicago as well as at NIH National Eye Institute. Then she worked for several years in life care innovation, and currently she is a clinical research consultant. And for moderating the interaction session, I would like to invite Dr. Amir Nazir.
Good afternoon. We are constrained with time, so we will make this session rather quick and short. And thanks to all the panelists for joining us. We had some students who actually reached out to us that they would want to interact with alumni, and they might have questions. It could be about their career progression, it could be career choices, it could be whatever challenges they face. So raise your hand, students who have questions, and then I would point at you and you will have to go ahead. So I have Astha there. Astha, please go ahead with your question. So team, could you please send a microphone? Okay, microphone is already there.
Good morning, all of you. Myself, Astha, working as a senior research fellow in pharmacology division. So it's my great pleasure to attend this alumni meet. And I want to ask all you in general that I am a 5th year PhD student, and now I am progressing towards that whether I have to choose in the academy or the industry. So I want to ask your experience about it, that how should we decide, and as there is lot of immigrations issues related to the US visit or US transition. So I want to ask you that as a student, what should we consider about the choosing our decision wisely?
Who wants to take that question? Chitra, yeah, Ranjana ma'am? Yeah. Okay. Ranjana ma'am, you want to take it, or Chitra ma'am can take it? Any of you? Yeah, yeah, sure.
Yeah. So thanks, Astha, and welcome to our alumni group. Your question is where I should go after this. See, if you ask me and my husband Pankaj, we will say US is not the ultimate place. right? So unfortunately or unfortunately, we are US citizen but still living in India. So it's not an ultimate place. Secondly, after PhD, it's your decision whether you want to go to industry or you want to go to academia. You can pursue in academia as well, but if you want to go to industry, then आपको crowd से हटके चलो. You should take another course or another diploma or study for anything else. Are you from pharmacy background or what?
Pharmacology.
No, but pharmacology after MSc
Masters
or
किसमें?
after master?
So masters in zoology. MSc. She's in MSc.
MSc. Okay. So it's like if you want to go to industry, you can opt for management first. Because industry में survival के लिए तुम्हारे पास enough है. ऐसा नहीं है तुम्हें एकेडमिया में तो बहुत ही मतलब नॉर्मल पर्सन भी एंटर करके एक्सीड कर जाएगा बट इफ यू वांट टू बी इन इंडस्ट्री यू हैव टू बी रियल स्मार्ट to survive in any industry.
Wonderful.
Right?
Thank you so much. Thank you, ma'am, for that
So-
perspective. I saw Ashtubuja's hand also up. Ashtubuja, please. Yeah. So,
Sorry, I forgot the name. So I'll add Astha. So Astha, I'll give you a perspective. Will lot of my journey. So I did my PhD. I was maybe in that floor and multiple building. I think I was the one which was the lowest in terms of aptitude. But this institute gives you not only just the credibility and capability to do research and publications or do drug discovery, but it gives you the opportunity to understand the ecosystem, which we have to now, as a student, you have to start understanding. Just understand your ecosystem, not only for the job, but if you want to do job, understand what is the job ecosystem. At our time, there were hardly any jobs in the industry because pharma industry, it was very small industry. There were very few jobs. Chemistry department used to get job very easily, pharmacology and all, not unlike others. But right now, the ecosystem has changed. It's entirely flipped now. The opportunities for job is enormous. You have seen a presentation in the morning on CDMO. The CDMO ecosystem has spurred like anything in the country. And CDMOs are the one which are having the highest advanced ecosystem. And because they are working, they're serving mostly the external clients, like the gentleman used the word Novartis, right? All of us, all the multinational companies are dependent on CDMOs because nobody is doing the entire end-to-end research in their own company. Second, the jobs are there, multiple categories of jobs are there. So that was not existing that time. It's just you have to start understanding what kind of jobs are there, how to understand what kind of jobs are there, and how to go and apply for that. Because this ecosystem is little weak in our case, unlike IT and other industries where you have a site and you get all the jobs. And second is, in terms of career, like I have done my PhD, then postdoctorate, but I understood I don't want to pursue anymore in the core science, but I'll be around the science because the purpose was to serve the healthcare sector because I always feel a strong urge from inside in the public health. So and second, I always visualized myself as a corporate person. In somewhere, I dreamed myself as a corporate person. So I got two opportunities again after completing my postdoc, which was postdoc was also in India. And from National Institute of Immunology, I got two opportunities. The one which was the corporate job, and another was again continuing in some other institution. My job which I got was very, very small salary. My boss was very unhappy, but I took the risk. And when I came back out of that organization after 5 years, I had my salary 10 times bigger than what I joined. Then my boss was happy. Then I joined another side of the industry. Which was, I also understood, and that was accidental. And I went into Confederation of Indian Industry, which sometimes is perceived as a event management organization. But what we provide is the platform for industry to come together and discuss their issues with the government and transform the policy, which then impact all of us, either into the research institutions or the CDMOs, like the gentleman in the morning was saying that now they have started an association of CDMO. I know, and I know they're trying for last 3 years, and I was working with them also. So we do a lot of policy works, and that gave me another opportunity to now contribute to overall health sector from the policy side. Where
Yeah.
I'll give you the example: if a drug of 1 rupee is available in the company, but it is not available to the patient, that's the access issue, and that's where comes the policy. So a lot of angles to the policy. So the opportunities are enormous. CDRI, because of CDRI, unfortunately or fortunately, I never failed anybody.
Wonderful.
Because we are given the overall courage to take the challenges and conquer those challenges. So far, I have worked in 2 multinationals, in one US and India strategic forum, where I was supporting a lot of US companies also, and I have never failed. And fortunately, this is all because of the credit goes to the people who have given that courage and strength to take up anything, whatever comes to us.
Thank you
So
so much,
don't be
Dr.
worried
Gupta.
about the industry.
Thank you so much. Those are really interesting perspectives. What we'll also do is, wherever students you see an answer is coming for you, since we are like constrained with time, even if an answer is short here, please catch hold of your alumni seniors and have further discussion even after this. Meet them during the lunchtime and make use of this. Not only these panelists who are here, if you see that there would be someone else in the audience who can help you, please do connect. And so in the interest of time, we will keep your questions and answers also rather quick. So I will go to Ashtabhuja now for his question, and then we'll move on. Yeah.
Good afternoon, everyone. I am Ashtabhuja Mishra from MPC division. My question is kind of similar to Astha's question, but I am being very specific to industry. We, as a chemistry scholars, we used to go for industry and postdoc, mostly in USA. But now, because of fund and visa issues in USA, the opportunities there becomes huge crowd inside the opportunity inside our country. So in this situation, what we should do? Because usually when we complete our PhD, we used to get jobs in industry, but nowadays it becomes difficult because of the crowd. So what should we do?
So Dr. Srinivas is perfect person for this. Yeah.
Yeah. So good morning, all. So I heard about your question. So it is not the matter of getting job. So this is the institute where you are living is a big paradise. You have all opportunities to carry and succeed in your career. From this is the basement to think beyond your expectation. So what you are expecting at the moment is you want to enter into industry or you want to enter into academy as a postdoc or an employee. But this is not sufficient to think enough. You have to think beyond this. If you think you want to become an entrepreneur, then there is no question of thinking of getting job. So there is not at all required, right? So what I am suggesting, while doing PhD, you need to excel in every segment. So usually a medicinal chemist working in the laboratory and getting knowledge and carrying the things in that narrow direction. But that narrow direction is not at all sufficient to survive and sustain in this competitive world. So you have to adopt all other areas what you have in CDRI. So CDRI is the only institute having enormous number of departments and all kinds of facilities. People are using a limited resources like only synthetic abilities they are obtaining and only HPLC, LCMS, and the nearby systems they are adopting and they are excelling and going away. But apart from that, if you learn about the computational side, if you learn about docking side, if you learn about other biopharmaceutical side in the same institute, then you will learn things wherever you go in postdoc or in industry, and you can adopt the techniques instantly and you will excel in the field.
Thank you,
So
Dr.
there is
Thanks.
no possibility for falling down, and you will not lose anywhere. You will get the job, and you will give the job for sure.
Wonderful. Dr. Srinivas is a perfect success story for all of you, Ashtabhuja and others from chemistry. Even after this meeting, please contact him. He has set up a wonderful lab, Dr. NS Labs, and one of the purpose of this alumni meet again to get CDRI together with their alumni. We are already doing a project with them. In fact, couple of those we already completed, and currently also we have a project going on with them where we are doing some synthesis of one of our lead molecules, which is going for IND enabling studies, and we had to do scale up, and Dr. NS Labs came forward and the order went to them. So he's a perfect success story for you. Please contact him, take his contact information, and stay in touch with him to learn further from him. I saw Anisa's hand up here. Anisa, next question from you, please. Be very specific and quick. Yeah.
Good afternoon, everyone. Myself, Anisa Noor, and I am currently pursuing PhD in medicinal chemistry. So, as a PhD student in medicinal chemistry, I see career paths largely oriented towards traditional academia or pharmaceutical industry. From your experience, could you please share some insights about some lesser explored or lesser explorer regions where a PhD student can make meaningful impact?
Manish, you want to take that up? Yeah, please.
I am the perfect example for that.
Yes.
Chal raha hai? Ma'am,
Okay, so I did medicinal chemistry here, and then I moved postdoc. And luckily, just like you say, I'm also struggling to get a postdoc. It's not easy. We file 100 application per day, and then we are getting nothing output out. Then I got a postdoc position in France. So that postdoc position is for the nuclear chemistry. I don't know anything about the nuclear chemistry. Even nobody in CDRI also know about it. So I talked to Dr. Goel, what can I do? Okay, तुम्हारे हाथ में कुछ नहीं है, go ahead. I landed there, and there is a big problem of language. Even if you want to withdraw money from bank, you have to take somebody for you. I fed up with the system because with such much of knowledge, you are learning French. And my PhD supervisor is so happy that okay there, but my postdoc supervisor is also very good. After the review meeting, he explained in English. Then I thought it's a very stupid thing. What they are speaking in French, and then he is just narrating me. I quit the job, and then I landed in the US with the basic chemistry, medicinal chemistry job. After there also, because what we learn in CDRI, that's hard, never failed. I made a molecule which they are struggling to get from last 5 years, and we finishes in just a month. So after that, he is very happy because he is the Lawrence Hurley is a G quadruplet structure. He designed the G quadruplet, proposed a G quadruplet DNA structure, and then again got a job in NCI, and again the same situation. That is also a radiochemistry job. That job I referred through Dr. Diwan Singh because one of his fellow is there, Mahen Nasser. Then I again talked to Dr. Goel. What can I do? Again, this radiochemistry jobs comes to me. He say, okay, maybe your destiny is that he's always forcing you to go to radiochemistry. I picked up that one. When I landed in NCI, you will surprise that there's no lab. Inside the lab, my supervisor is also from India. He did his PhD from IIT Kharagpur, and he did postdoc in one of the Nobel laureate there. He said, this is the lab, and we have to build it. And the bats are hanging there. And then we started the scratching because, okay, that is not the India. In next 6 months, we developed a radiopharmacy GMP certified lab there. That is the biggest challenge for me to develop that lab. It has everything from zero to human injection certification laboratories. It's an advanced GMP certification lab. So we established that one, and we transferred the 3 molecules from that lab to clinic in US. And one of the molecule is last year is FDA approved. That is the 18F fluoroestradiol for estrogen receptor imaging. And okay, I do not have any publication from that group, but I have a couple of patents for that, and also I have got the experience of setting a GMP lab in that domain.
Wonderful.
Nobody at that time in India is doing that kind of job. Then all of a sudden I got a job in SGPJ that they need a person who has MSc chemistry with experience in radio pharmacy and radio chemistry. I said, this is the perfect job for me.
Destined.
So, and that job was created by SGPJ after 7 years of gap. Because 7 years before there is a fellow there, he expired during the job and the job is vacant. And there is no demand in India. So that's why that job is still hanging on. So Dr. Gambhir has reproposed the job. And since they are planning to set up a cyclotron, that is a small reactor to produce the radionuclide. So only the radionuclide small reactor is available in AIMS and PGI Chandigarh and other the BRC, what you say about it. And he, then my application was rejected. Clerk say this will not accept because it came late. But as per the institute rule, any who is applying outside have 10 days extra. Then I have to greet the postman of the SGPJ. Okay, submit my application again. And I talked to the director and he agreed. And finally I was selected. And again when I landed, there is nothing in lab. Only cyclotron is establishing by the Japanese engineer and nothing, even a small spatula is not there. But I am lucky that my supervisor is so helpful in everywhere, whether it's PhD or in postdoc or here also. He helped me a lot. And I finally...
Is Dr. Atul Goyal here?
Yeah, he is.
Yeah.
So I established 100 CR facility in SGPJ. And I am proud to say that currently our facility is the only facility in India who is producing largest number of radionuclide and radioisotopes.
Truly inspiring story, Dr. Manish.
And
truly inspiring.
I never stop. I never run cyclotron in US. I talked to Atul ji again. Sir, मुझे तो कभी ये चलाना नहीं आता. बोले यार देख लेना जो होगा देखा जाएगा. I talked to Gambhir also, Dr. Gambhir. Manish, engineers will help you out. So we have very sophisticated facility. I think Dr. Saxena also visited our lab. Lot of CDRI fellow visited. Even recently, we finished 3 different projects from US company. One from Bristol Myers Squibb, one from Boy. We transfer technology. We develop one tracer for prostate imaging here and transfer technology to the pole atom of Poland. And they are trying another because we finished the clinical trial. So I never stop it. And recently, from last CDRI, I have started a collaboration with Zydus Life Sciences recently. Because what we do, we are labeling monoclonal antibody, we are labeling peptide, we are doing everything. And they listen my talk. It's just a 10 minutes flash talk. I got it here because still people are not understanding what radiopharmacy can do for them. But yeah, in US and China in last 4 years, lot of changes are done. What you say, what he said. Industry is changing very dynamically. We have to understand. Even lot of people from here, MSc chemistry or PhD chemistry, then go for nuclear chemistry. Lot of jobs are still vacant in US. Apply for the postdocs where this kind of applications are there. Because they are not getting people like you. Because we have...
Thank you so much,
After
Dr. Manish.
my resignation from that institute, that post is still vacant. So people
anybody who has expertise in this field may probably take expertise.
PhD chemistry guy can go for that.
Yeah. Exactly.
Yes,
that's what I'm saying.
I can help you.
So, so please, yeah, any of you, again, as I said, so even after this session, please contact Dr. Manish Dixit and whatever guidance you need in your career also. So please keep it. So in the interest of time, we'll move to next question. I saw Anam's hand up on this side. So please go ahead with your question. Be very specific and brief. Yeah.
Good afternoon, everyone. Myself, Anam Nasir from Division of Toxicology. I recently completed my PhD and my question is oriented to academics. So I wish to pursue postdoc in India. I mean, there are multiple national postdoctoral fellowships as well as INSPIRE faculty programs. But I recently submitted also, but my proposal got rejected. So I wanted to know what are the key points while writing a project proposal or choosing a PI for postdoc in India and while choosing the institute.
You need a connection.
Yeah. Dr. Srivastava would want to take that. Yeah, please.
I am the. I think in this panel, I am the worst person to answer this question.
Okay.
I never did the postdoc.
You need a connection, first of all.
I,
Yeah.
yeah.
Yeah,
but
yeah. Dr.
I will
Batra
try
is
to answer
sits.
your
Yeah.
question in a way. See, when we complete our PhD, when we come to the CDRI, I'm taking the specific example of the CDRI. We have one goal in the mind. We have to do PhD, nothing else. Whether it is 3 years, 4 years, 5 years, but the objective is PhD. When we are on the verge of completion, like Astha or Ashboja or these questions, they are connected with the future. And the easiest future which is looking is the postdoc. We don't look what we are going to do. We just focus, okay, I have to get a postdoc. And probably postdoc, nowadays people are doing in India also, but earlier in my times, I'm a pre-2000 era, so 99 I moved out from the CDRI. So that time postdoc means going abroad, primarily US or to the Europe. But see, in my mind, you have to see in the next 2 years, 3 years, 4 years down the line what you want to learn or achieve. Whether it is coming through postdoc, through academics, through a job, or through anything. I got a postdoc, but I did not go to the US. I decided to stay back, and my mentor supervisor, Dr. Katti, he called my father to the institute and said, this fellow is going to spoil his career. This has gone mad, kind of the words. I'm not having the exact words, but similar to that. And my father also tried to say counsel me or brainwash me. But I decided, no, I don't want to go there. I want to be here in the industry. I came from MSc chemistry background, went to the division of biopolymers, working on the nucleotide, peptide, although it was a chemistry, but something different, more connected with the DNA and protein interactions. When I moved to industry, again, I found some of my CDRI alumni who brought me to the core chemistry, which today I call, the fancy word is the process chemistry, but we call it as a mistrigiri of the chemistry, which is fine-tuning the chemistry, optimizing the chemistry to get a better and cost-effective and a simplified processes. And I was carried away with the not doing the postdoc, going into the job, then changing the jobs, initially a couple of times for my own benefits. Then some other direction I took from the research to the research management and then core commercials. Then I entered into a field of entrepreneurship, which is a big buzzword. But believe me, there is no such word called entrepreneurship. They come with the horns or something. I'm sitting here, I think, enjoying with most of the people who have that entrepreneurship skill. That is from the days of the CDRI. It is not there that we got it after 10 years or 20 years. And I give you one very quick small example. In our laboratory, when we were preparing nucleotides, we were to have the very hygroscopic products to filter those at creating nitrogen atmosphere and all that. There was Mr. Chopra in the glass blowing section. Some old people must be knowing. He put a sintered funnel on a glass flask for us so that we can directly filter it from the glass flask without opening it. This was the innovation, or the, I will say, entrepreneurship skills of mine as well, Dr. Katti, Mr. Chopra, who helped it. So I'll suggest, look at the science. Look at your vision for the next 4-5 years, not 20 years, 25 years. What you want to achieve, whether it will be fulfilled through a postdoc, go for a postdoc. If it is your vision is to go to the US, go to US. Then don't look at which lab, which work you are going to do. Make your priorities. What is your first vision, then second, then third, and then accordingly go. And sometimes you will find the different signs in your life. If you identify a right sign, turn at that time. You will not regret. You will never regret. That's my experience. I can share. Thank you.
In the interest of time, we'll take some more questions. And what I would recommend for Anam, meet Chitra ma'am also, and Dr. Batra, serves on all of these ANRF and DST related committees. So he will give you a perspective as well. So please keep interacting after this. Can I have a show of hands again? So for last question. So this girl here, yeah, please.
Good afternoon to one and all present here. So my question is a kind of extension from Astha ma'am's question. So basically, there are two points in our life where we have to decide things. One is after PhD, one is after masters also. So my question is purely on the basis of industrial aspect. Uh, like when we do our masters or when we do our PhD, it is not necessarily that we work on analytical instruments like HPLC, GC, LC, MS, but the industry demand that kind of thing. Sometimes we are just into in vitro things or in vivo things in our whole PhD or masters. We don't acquire that particular skill trait which are needed for industry. So how can we go to the industry? How does this particular thing will help us, or whether we have to take whether we have to acquire those skills also apart from what we are good at.
Thank you so much. Any one of the panelists can take it. This is the last question.
Confidence is very important. Actually, I never run cyclotron, but I have a confidence I can do it. So it is not a instrument. It is you run the instrument. Instruments are designed in such a way that end user can easily handle it and run it. So if that is so complicated, it never comes to the market. That means instrument, you have to learn the technology or concept. If you know the concept, you can run any equipment. That's not. So before you start any equipment, you have to learn the concept of technology of the equipment use. That's all. If any equipment, you can run it. Maybe you have some flaws. Okay, fine. But as you learn, because initially a kid is crawling, then he start jumping and running. So everybody, same thing for you also. It doesn't matter whether you ever learn LCMS or not. Right? So I have a 37 different equipment in my life. I never run 37, but I run now. So it's not a matter.
Thank you so much. One last question. So again, show of hands here, Gaurav. Please.
Good afternoon, everyone. Myself is Gaurav Kumar Gupta from Division of Toxicology and Experimental Medicine. I am working here as a study personnel in GLP and non-GLP studies. May I ask the career opportunity for master student rather than CROs?
So his question is career options other than CROs for a person who is trained in GLP. So Dr. Srivastava wants to take this, please.
Again, I'll try to deflect the main question and answer whatever I want to answer. So excuse me for that. But see, whatever is your training, that training is for something which you got in these few years. Hello?
You're audible. You're audible. Yeah.
Don't just stick with your training that you want to pursue exactly the same thing. Look for the opportunities where you can do excel. You have the right example. What he was trained at, he is not doing the same thing. So this training is basically you got some skills. Now you have to look for the tools which can sharpen your skills. That's full stop. So you will have enormous opportunity. In the last couple of days, yesterday and today, when we came to the CDRI, a lot of us came from a different backgrounds, different colleges, different streams. We were here for some time, in different period of time, of course. Now again, when we met, we came from a very, very different backgrounds, doing a very, very different kind of the work. So don't just limit yourself whether you are GLP or something, and just before you, somebody asked the question, what industry is looking from you? We are looking raw talent, nothing else. Anybody, even in the CDRI, when people, I think Dr. Dikshit and Dr. Batra can confirm this, when you were inviting the PhD students to the CDRI, what you were looking? You were just looking raw talent, nothing extra. So everybody is looking for the same. You also look for the same. Don't just look what you have learned and you want to extrapolate that.
Thank you, Dr. Srivastava. I'm sure there are many questions and many answers also from the panelists. So I would strongly encourage all of you to please interact with the panelists during the lunch hour also. Take their email IDs, phone numbers, WhatsApp numbers, stay in touch, learn more, and do not end this here only, and keep this conversation going. And whatever questions you have, I'm sure they will be happy to help. Thank you so much. Thanks to the panelists for your time. In the interest of time, we'll have to cut it short, but please stay in touch, and thanks for your wonderful perspectives.
Hello, good afternoon once again. Now, so I'm sure all the students must have heard the opinion of all the panelists, and I'm sure you must be getting benefited out of all those opinion and the suggestion that our panelist has shared with you. So now we have come towards the end of this session, and I would now request our chairperson, Dr. Amir Najir, to felicitate our panelist. Thank you to all the panelists, and I now request all of them to please be seated in the auditorium. And very soon we will begin our next session. So in 1 or 2 minutes, okay. Sir. So now,
yes, conclude.
Okay. Yeah. So moving towards our closing ceremony session, I request our director, ma'am, Dr. Radha Rangarajan, to please come on the stage. And Dr. Amir Najir and Dr. Vinita Tripathi, chair and co-chair of Alumni Day. So please come on the stage. I would now call upon Dr. Sanjeev Yadav and Dr. Hizaz for convener and co-convener to please be seated on the dais. So with all dignitaries on the dais, I now invite our respected director, ma'am, Dr. Radha Rangarajan, to kindly deliver a closing remark and formally conclude this academic proceeding of this alumni session. Ma'am, please.
Thank you, Kinshuk. I think a lot has been said, a lot has been discussed. I don't want to spend too much more time talking and being between you and lunch. So I just want to say that it's been a really fantastic couple of days. There have been so many incredible conversations, new openings, possibilities of working together opening up, so many suggestions, so many words of appreciation said. I think for all of us who worked on this particular event, it's been fantastic to hear that the alumni, all of you being here, was such a delightful experience for you. The teams have worked very, very hard. I want to acknowledge the fact that they really wanted, they put in so much effort so that the experience would be as warm and memorable as possible. So with those few words, I think my sense is that we have opened a new avenue now for alumni to be closely aligned with the institute, to interact in some formal way, and I look forward to taking some of the conversations forward and creating real connections with the alumni, between faculty and alumni, students, and everybody else who's currently at CDRI. The one thing I'll say is that I request all the alumni to go back and speak to their friends. Please spread the word. Please let people know about the Alumni Association. Please encourage people to become members. The second thing I'll say is that we will create a compendium of some sort in which we capture alumni interests and specific areas that they would like to contribute to, so that we can start to build some sort of a matching system whereby we can connect on specific projects or queries or seek specific kind of help. And the third thing we'll do is that we'll try to create some sort of a alumni talk series or alumni engagement series where students can interact with one particular alumnus or a set of alumni together, but it gives time for more structured engagement. So these three things we'll do right off the bat. Additional suggestions are most welcome, and as the association becomes more formalized, I'm sure there'll be many more things that happen. If you think of any other ways in which you would like to support or be part of CDRI, please write to me anytime. On our website, perhaps we'll create a tab or a link so that you can easily reach us. I think this is something that we need to add. Is the website committee anybody here? Santosh, yeah, please make a note so that we can improve alumni engagement with CDRI. So with those words, I wish you all the very best and great, a safe journey back to your homes. Lots of new memories, no doubt, have been created. We hope to see you back again in the near future. We'll try to do this as an annual event. Thank you once again for taking the time to be here.
Thank you very much, ma'am, for the closing remark and emphasizing one important aspect, that we have to stay connected so that we can nurture the relationship of alumni versus faculty member and the student, and that will be very, very helpful. So with that, now moving forward, I would like to invite the students who has done fantastic job in terms of the poster presentation. We have 4 awardees and there is no ranking because all of them did a wonderful job. So I will call upon the students. The first student, Vishal Kumar, then Rahul Baghel, then Astha Singh and Nikita Chikara to please come on the stage. And I request director ma'am to felicitate them. So Navjyoti Scientific has offered a very good help, that they have given the cash prize money, and that we are distributing 5,000 rupees as a cash prize to all these poster awardees. 5,000 each. Yes. Thank you, ma'am. So now in next sequence, since poster judges has done a lot of fantastic job to identify the candidates, and as a token of appreciation, I would like to call upon the poster judges, Dr. Prabodh Kapoor, Dr. Parul Misra, Dr. Abhay Tripathi, and Dr. Rashmi Singh to please come on the stage.
Parul left. I don't know about. I think one of the judges is here.
Yeah. Okay.
Okay, Prabodh and Parul.
Thank you, sir. Thank you, sir. Now I request dignitary to please take their seat. And I would now like to invite Dr. Hizaz to convey the word of thanks.
Good afternoon, everybody. I feel proud and honored and privileged to be part of the front-running team which made this alumni meet possible. I'll start with a short story. This is a group of people, maybe a research group. A research group was going out for a lunch, maybe for celebrating their a recent paper published. So the PI also take her 6-year-old son along with the group. So when they are in the town, they were a bit confused about which restaurant to go. We may go here or we may go there. So this 6-year-old boy, he told, Mama, let us go to this particular restaurant. Then his mother told him that is not to you, it is to the team to decide where to go. Then the boy asked, what if the team decides wrong? Should we still follow them? She replied, that is why you need to be wise to choose your team. So, I believe we were wise to choose our team, and we had a great team, and that great team put out this great show for you yesterday and today. So thank you so much to the whole team. And I'll start thanking DG CSIR, Dr. N. Kalaiselvi, for sending her recorded inaugural address for us. And also our guest of honor, Dr. Ram Vishwakarma, a distinguished scientist, CSIR-CDRI, for coming and gracing our stage. He spent the whole day with us yesterday, and it really means how much he cares to his alma mater. We also heard it from his speech that he gave yesterday. And so before I move on with the formal vote of thanks, I would like to brief about the preparations that we did towards this event that you all witnessed yesterday and today. So 2 years ago, when I joined CDRI in our first one-to-one meeting with Director CDRI, ma'am, if you remember, one thing that you mentioned as one of my responsibility was to work for the Alumni Connect. She asked me to go and talk to my senior colleague, Dr. Sanjeev Yadav, and I did so, and he was already working with it. He had a good database, and he was already connected with many alumni. And then he told we may make WhatsApp groups and connect with alumni, and also this can be done so that we can slowly build on that. And we did exactly the same. We created WhatsApp groups, different batchways. Most of you will be there in different groups, I believe. I also see a spike in the number of members in different WhatsApp groups yesterday. A lot of people was coming there. And so we did so, and we started the preparation. We started to engage with alumni. And when the 75th year celebration started, Director CDRI told us that we should have an alumni meet. Maybe it would be much better if we also have an alumni association, if we can facilitate the formation of an alumni association. So this alumni association, an alumni meet that we can do in CDRI, but forming an alumni association, we need it's a legal entity. We need people who are really enthusiastic about it to do so. And luckily, Dr. Batra got retired in last September because that was previously stopping him forming the association. He is really enthusiastic about it. He was trying it earlier, but since he was an employee, he couldn't do it. But for our luck, he got retired, and he was really enthusiastic on this. I still remember, sir, your last day of retirement. If you remember it, we both were me and Dr. Sanjeev Yadav were the last two people. We went to his office for an official discussion. So it was at evening 6 p.m., the last working day of Dr. Sanjay Batra in CDRI. So at 6 p.m. when we went there, he was actually giving out things to different colleagues in the department. Somebody was there. He was taking his computer. So so on the empty table, we discussed and we handed over him the papers, the draft that we prepared, which may help him in forming the alumni association. So on that day, on the day of his retirement, he started, and at that moment he started working on it, and he went on to his colleague, he spoke to them, he convinced them, and they were all on board, and they formed an alumni association, which we never had in the last 75 years. It's a very big thing. And so I thank him a lot. I congratulate him a lot for doing that achievement. And I also thank all the founding members of the Alumni Association, Dr. Madhu Dikshit, President, Dr. W. Huck, Vice President, again Dr. Sanjay Batra, Secretary, Dr. V.L. Sharma, Dr. K.K. Srivastava, Dr. Shailja Bhattacharya, and Dr. Archana Telwar. So they made it possible. They made the impossible possible, basically. And this happened because we have our visionary director with us. As I just explained, we started preparing for this 2 years ago, and it is my case. It's after I joined. Of course, much work was done before that too, and I'm sure the thought process started. Director CDRI has started the thought process much before that, and it is her administrative support, her enthusiasm, her decision that this should happen, which made this possible. So thank you so much, ma'am. Your determination made this possible for us. And in a recent meeting, one of the preparatory meeting, she told that Alumni Association is one engine for the growth of another engine for the growth of the institution, and CDRI was lacking it. And because of her determination, now we have that engine. I'm sure this engine will work efficiently and smoothly, rigorously in the coming years for CDRI. And now I have to mention two names here, two big names, Dr. Amir Nasir and Dr. Vinitha Tripathi. They are the chair and co-chair of our 75-year celebrations. We were under their shadow. We were hiding behind them. They were taking all the responsibilities. They were taking the responsibilities of not only the right thing that we do, they were also taking the responsibilities of the wrong thing that we do, which gave us a lot of courage, a lot of confidence that we can make mistake, they will save us. So thank you so much for being there with us, for both of you. And they were not just the chair and co-chair, they were with us everywhere. Dr. Vinitha with us even in the town for buying the registration kit. So everything what you see here is possible. In the last 2 days was possible just because of these two people, Dr. Amir Nasir and Dr. Vinitha Tripathi. And then I thank all the participants, all the alumni who decided to come here. It is not easy to attend an alumni meet, because last year there was an alumni meet in my institute, and I did Trichy. It was in Chennai. I thought it is very difficult to go. I didn't go because I thought I need to take leave. I need to take flight tickets, and I couldn't make it. But you guys made it. It is really wonderful. You all really deserve a clap for that. Thank you so much for coming. Thank you so much all to all the speakers, to the session chairs, session co-chairs, all of them, to the judges, to the panelists. Thank you so much. And then from the CDRI part to our team. So I'll start with our honorable COFA, Mr. Sanjeev Shekhar. He has helped, I think he's not here. He has helped us to navigate through the red tape. He made our path easy. And of course, we would need his help in the coming days as well. And definitely. And also head PME, Dr. Anand P. Kulkarni. He also helped us in many things. He has guided us from the beginning onwards of this event, and also facilitated the registration process for the internal members. And similarly to the senior COA, Mr. Bhaskar Jyoti Diori and his office. Thank you so much for all of them. And now, when coming to the committee, before coming to the committee, I would like to thank to Dr. Arnav Das Gupta. His name you cannot find in the listed committee members, but when he stepped in, he made our life much more easier. He helped us in many ways, and you have no idea how much you helped in this. I'll tell you later. Thank you so much. And then Dr. Anil Gaikwad, it's also a big name for this event. And I have made a lot of troubles for him, but he engaged in everything which we requested. Thank you so much. And then all the session coordinators, especially to Dr. Shruti R. Raju and Dr. Kinshuk, they have did a lot of homework, and they pull off the spectacular show that you have seen here. It means a lot of hard work that both of them has did. Of course, they both are professional anchors, but it means a lot to us. And same goes with the other session coordinators, Dr. Namrata Rastogi, Dr. Smriti Badoria, Dr. Nidhi Kumar. She herself called Nidhi Ayog yesterday. I'm sure that is a favorite session for all the alumni, right? It was, yes. She did a lot of homework. She did a lot of homework on that. And Dr. Rajdeep Guha and Dr. Sachin Kumar and Dr. Ajay K. Srivastava. Dr. Ajay's name is also a very big name. He helped us a lot again. So whatever we requested, he made possible. He is not in the hall now. We are missing him. But Dr. Ajay has done a lot, many things for us. And then Dr. Shabir Ali. And again, when I mentioned Shabir, I'm not sure he's in the hall. Shabir, I didn't allow him to sleep last 1 week at least. He he did a lot of things. He take care of the registration committee. He took care of the poster coordination and many other things. He's yeah. Shabir is a gem that CDRI has, and yeah, I hope I won't miss any names here to mention. And then when I go by, I'll go by the committees now. So program committee, actually Dr. Ridu Trivedi is the program committee coordinator, but she couldn't join us during the event because of some health issues. But thanks to her for her support, and Dr. Namrata Rastogi and other members in the program committee. I'm not reading all the members because it will take a lot of time. And then again, Shabir's name is there in many columns here. And then again, registration and reception committee, Dr. Divya Singh, Dr. Smriti Badoria, and Anubam Jain. Anubam Jain has contributed a lot to the committee, and Piyush Kumar Gupta. So Piyush was started to involve in these activities in the last moments, maybe in the last week. But before that, the last 2 months at least, academic affairs was running smoothly as it was earlier. And it was because of Piyush. So he take care of everything so that we were free. Piyush is another gem that CDRI has. And to the whole team in the academic affairs, and Dr. Shiba Saji Samuel, once again, big kudos to her. She took care of the registration committee. And then the cultural committee, again, Dr. Ajay and Dr. Ravi Shankar and their team, and all the alumni employees of CDRI. They helped us regarding the Alumni Connect. And Dr. Kishore Mohanan and Dr. Anil Kumar KS for transport and accommodation. Actually, it's not a big deal for them. After doing CTDDR, these kind of events are peanuts for him. So, yeah, they are. They can easily, very easily handle all these things. And so many times when I speak him about the transport, he says, "Yours is a very small event. Forget it. I'll handle." And Dr. Anil Kumar KS, again, it's a big name. He takes all responsibilities himself. And their team. And then again, the campus adornment. Again, it's all Dr. Vinita Tripathi. And yeah, all the floral marvel that you are seeing, it's all come from her. And of course, many people helped her. And then I mean there are a lot of people in the list, so I'm not telling their name. It's all her, yeah. So, and then the printing committee, again Dr. Anand P. Kulkarni and his team. Again, Shabir was there coordinating things with him. Dr. Anand P. Kulkarni has also done a great job here because we were almost on the edge of time, and he made it possible for us to get everything printed before time. And when I speak about abstract book, he has gone through the abstract book line by line along with Shabir. So thank you so much for him too. And again, the auditorium team, Dr. Santosh Kumar and his team, Sumit Kichi and the others. And the photography team, Mr. Ravindranath Londhe and Kazim Raza. Everybody knows the clap automatically comes here. Events goes in CDRI one by one, but these are the two people. They work tirelessly, endlessly. No Saturday, no Sunday, no night, no day. And again, both of them, big kudos to both of them, to Ravindranath and Kasim. And Kasim is another gem that CDRI has. I have noted 3 gems here: Shabir, Piyush, and Kasim. This is deliberate. And then I have already mentioned Dr. Anil Gekwa, Team Food and Catering, and his team. Thank you so much, sir. And Campus Tour, Rajdeep Guha and Dr. Sachin Kumar. I'm missing doctor somewhere, please don't mind. Dr. Rajdeep Guha and Dr. Sachin Kumar. And then a sponsorship team, again, Dr. Ajay K. Srivastava, Dr. Naseem Ahmad Siddiqui, and our treasurer, Dr. Agilesh K. Tamrakar. and all those. And again, Mr. Sanjeev Shekhar there, he has given a lot of help regarding handling the advertisements and how to get the money in account and all. Thank you so much to him again. And then Prasan Media, which was handled by our own team. I will not congratulate anybody. It is us. And then website, engineer Kudal Srivastava and engineer Santosh Shukla and their team. Thank you so much, sir. Thank you so much. We have bothered you too much on the website. And we also launched a portal yesterday. All the portal was made by them, by their team. And again, they are another team. They work tirelessly here in CDRI for all the events, for everything happen. They have to give the IT. So that's always done by them. And there was a team, they were taking your interview bites. Ma'am, we also have that video. Shall we play that? Or if, yeah, okay. After the finishing this, we will play that. Sure. Yeah, I'll finish the vote of thanks. So, so they took your video. That was Rati Kumari and Bibhendu. They did a big job on the with the camera and the interview. And the finance and purchase committee all mentioned. I have all names I have already mentioned, so I'm not mentioning again. And security and medical, Dr. Ravi Shankar and his team, and Saurabh Singh. So yeah. Yeah. Okay, sure.
That's not ready. Don't forget kinship.
You're going to be the compare for
yes, rest of your yes. You're doing a wonderful job. Thank you so much.
Thank you.
Absolutely fantastic. Thanks. You continue with this, and then who shares now that names that do share a share. Okay, sure. So now we have 5 more minutes. Do you complete your vote of that? And then invite Dr. Agni is there. Dr. Agni. He wants to read 2 lines.
Okay. Yeah, so now we have a lot of time. So yes, actually I'm done, in fact, with the vote of thanks. And I think I did not miss any big names, but I didn't read out all the names. So I'm sorry for that. It's just in the interest of time. Again, I would like to mention once again Dr. Shruti Arraju and Dr. Kinshuk for all the outstanding hard work and the performance that they presented for us. Yeah, Shruti and Kinshuk, could you please come on stage? Shruti is not here. Okay, yeah. So that is it. So now, before I leave the stage, I'll make one more announcement. Actually, regarding the Transport and Accommodation Committee, their job is not yet done. They are still working. So we have the Chhatr Manzil visit, and we will start by 2:30 or 2:45. We will start by 2:45. So there are 2 buses plus 1 traveler arranged, which is right across the food stall. So through the poster station, like just behind this venue on the road, you can see 2 buses and 1 traveler arranged. And I would so this is mainly for the alumni who came. So of course CDRI students and staff can join us for the program, but the travel arrangements made are for the alumni. Okay, so that is it. So and now I will invite Dr. Agnihotri. He would like to conclude by a couple of lines this program. Thank you so much, everybody.
नमस्कार, आदाब। ये आदाब जो है सिर्फ लखनऊ की देन है, इसकी ओरिजिन लखनऊ में हुई है। जैसे कल लाल साहब बताए थे ना कि उन्होंने यहाँ से आप कहना सीखा, वैसे आदाब के बारे में ये है। मैं यहाँ पर 1984 में आया था। उस समय CDRI 33 साल पुराना था, आज 75 साल पूरे होने जा रहे हैं। मैं एक शेर कहना चाहूँगा, ये हकीकत बहुत पुरानी है, यानी कि करीब 41 साल पुरानी है। ये हकीकत बहुत पुरानी है, मेरा दिल CDRI की राजधानी है। ये हकीकत बहुत पुरानी है कि मेरा दिल CDRI की राजधानी है। हम इस पर क्यों ना करें गुमान, हम इस पर क्यों ना करें गुमान। यहाँ से जुड़ी हर किसी की कोई ना कोई कहानी है, यहाँ से जुड़ी हर किसी की कोई ना कोई कहानी। एक शेर मैं और अर्ज करना चाहता हूँ, समाप्त हुआ है। राहत इंदौरी साहब का शेर है, हम अपनी जान के दुश्मन को मेरी जान कहते हैं, हम अपने जान के दुश्मन को मेरी जान कहते हैं, वतन की इस मिट्टी को हिंदुस्तान कहते हैं, और देश के इस खूबसूरत संस्थान को औषधि अनुसंधान कहते हैं। देश के इस खूबसूरत संस्थान को औषधि अनुसंधान कहते हैं। थैंक यू वेरी मच।
Dr. Batra wants to make a quick comment. Yeah.
I think all the alumni members who are here will agree with me. All the effort made by Hijaz and Sanjeev, of course, they were guided by Amir, but
I actually came to say that only, and then he stole my show here. So I was actually going to request all of you to rise and give a standing ovation for two people, but in particular Sanjeev and Hijaz. Sir, ye main karne wala tha. Please. So they are the two people behind the show. All the groundwork, all the running around, and whatever you saw has been done by two of them. So of course there was support of all of us around, and then they probably have not rested or slept properly for the last at least 2 months. So thank you so much, Sanjeev. Thanks, Hijaz. And thank you, Dr. Batra.
One more thing, all alumni who wants to connect to CDRI or wish to give their name, email ID or a telephone number, they can write to me, they can contact Hijaz, Sanjeev, anybody, or means director will forward, of course, to them, or anybody, or anybody of all of us, all three of us, and we will take care of that their name is included, and we'll, of course, the website will be running, and we'll have a Google form where you can write things. Yep.
Have the list whatever we have right now on our website.
That we'll do.
And generate a Google form where everybody can send.
No, we'll do that. We'll do that on the website, and we'll do that.
Perfect.
Bilkul.
Perfect. Thank you so much, everybody. With this, we'll conclude the program. Let's rise for national anthem, and after that, we will disperse for lunch and then onward for the tour to Chhatar Manzil. Thank you so much. Please rise for national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे।
Thank you. Please join us for lunch, and we will start for Chhatar Manzil tour sharp at 2:45. Thank you.